Pasprazzo, your biology master teacher. So students, before we start today's class, quickly let me know in the chat right now if my voice is audible to all of you and the beautiful presentation behind me is visible to each and every one of you. Quickly let me know in the chat right now, all of you. Now students, all of you, right, you will be aware because of Navratri series. Because of the Navratri series, we are doing a special class right now. We are doing one shot of every single chapter. And that's right. Today is going to be a long class. We will be finishing the entire chapter of anatomy of flowering plants in the one shot now. One shot entire chapter. So with that being said, all of you raise your hands quickly. Everyone, quickly all of you raise your hands. Now, imagine, all of you quickly raise your hands. Imagine students, imagine. What if you remove the entire skin? What if you remove the entire skin and then what if you try to look at a hand? Obviously, if you remove the entire skin, will you be able to look at the blood vessels? Yes, sir. Will you be able to look at the bones? Will you be able to look at the tissue on the inside? Obviously. Now that is very fascinating, but also scary at the same time, right? It is very fascinating and also scary at the same time. But you cannot do that usually, right? You cannot go take someone's hand, cut it and then see inside. You will go to jail for that, okay? You will go to jail for that. Don't do all that. But the same experiment, you can do it for the plants. For example, tomorrow what you do is go out to your house, take a plant, pick a plant and try to cut it open. No one will put a case on you also. So take a plant or take a leaf, cut the leaf, open the leaf and look inside. Now, if you look inside the particular leaf or a stem or a root section and you keep it under microscope, and you will notice something like this. What is this? This is called as your internal structure. This is called as your internal structure of the plant. And students, let me tell you, disclaimer, okay? Can you see this? This one right here, this image, though this one is my favorite here. All of these sections were taken up by your friendly neighborhood botanist. So I was the person who took all this sectioning. So copyright alert, don't copy all this. Okay. So students, the whole point of showing you all this is when you look at a plant from the outside, it looks very beautiful, right? When you look at a plant from the outside, it looks very, very beautiful. But at the same time, you look at a plant from the inside, right? When you chop open a plant, right? When you cut it, when you take a sectioning, if you look inside of a plant, it looks even more beautiful. That is, look at this. Look at the sectioning of a plant. Can you see the stem section? It looks like a heart. This is a typical structure of a leaf. This is a root section, right? So when you look inside a plant, it is even more beautiful than the outside. And the study, right? The study of studying the plants on the inside, that is called as your anatomy. Anatomy is a branch of biology. Anatomy is a branch of biology which deals with internal structure of a plant. Internal structure of a plant. Students, is my voice not visible? Students, am I not audible? One second. Students. Some scam you're doing to me, I feel. Can you see that? I am audible only. Some scam. Oh my god. Yes. Full scam. Or oh god, full scam. I thought my voice is not audible. I have to go check again. Can't believe only, can't believe. So yes, I have completed this chapter before, but today is special because I'll be reading the lines of NCERT line by line. Okay, line by line. Okay. So that with that being said, anatomy is the internal structure. Now students, I'll ask you one simple question. Do you remember in the morphology? I spoke about some kind of modifications, root modification, leaf modification, root modification. Do you remember, all of you? Do you remember a stem modification, leaf modification, root modifications? Now you tell me, all of you tell me. If a plant is modifying from the outside, if a plant is modifying from the outside, will it also modify from the inside? Tell me, will it also modify from the inside? You're like, obviously, sir, I can't just change from the outside, I have to change also from the inside. Obviously, I have to change from the inside also. Now, why am I telling that? Because students, your entire plants, all the plants, almost all the plants, from the tallest of the trees, from the tallest of a tree to the submerged plants, the basic structure of a plant stem, root and leaf is same to same. Every single tissue, every single sectioning, if you take root, stem or leaf, it will have epidermis, it will have cortex, it will have a stele. So same to same in all the plants. Here and there a few changes will happen. That is because of adaptations. That is because of adaptations. Otherwise, it's always same on the inside. Same on the inside. That is, all the different plant parts are made up of same set of components. Just the arrangement is different, resulting in the different structure and different function. Now, students, tell me, did all of you clear your class 9th or did you fail in class 9th? Tell me, did all of you finish 9th class? Like, yes, you'll be like, yes, sir. 9th class, 100%. 9th class, 90% full flying colors. Okay. Now, students, I'll ask you a simple definition now. If you have passed your class 9th, I will ask you a simple, very simple definition. Okay. The simple definition is that, can anyone in the chat, or if you're watching it recorded, can you define a tissue? Can you define a tissue? Go. Your time starts now. Define what is a tissue. Like, what is the meaning of a tissue? If you have truly passed your class 9th, you will tell me what exactly is a tissue. Quickly. Your time starts now. Go. What is a tissue? You will tell me, sir, tissue is a group of cell. Simple definition. Class 9 student also will tell. Tissue is what? Tissue is nothing but a group of cells. Tissue is a group of cells. Like, okay, sir, group of cell. Okay. Now tell me, students, do they have a common origin? You're like, yes, sir. They will have a common origin. They will have a common origin. Beautiful. Now tell me, students, do they have a common function? Do a group of cells, when they're forming a tissue, do they have a common function? You'll be like, yes, sir. They will have a common function. They will have a common. Now, last one. Do you think they have a common structure? That's the important question. Do you think they will have a common structure? Tell me, students, yes or no? Do you think a cells, like a group of cells, do they have a common structure now? Common structure is there? Now, some of you will tell me, sir, in class 9th, my teacher told me, all of them have common structure. But right now, you're in class 11th or class 12 or class 13, whatever it is. Now, you can't tell the same definition for group of cells, tissues. Because in your class 11th, you study that. In your class 11th, you study that they can have a common structure. They can have a same structure, or they can also have dissimilar structure. What are you telling, sir? I won't believe you until you tell me examples. Yes, they can have a common structure. Now, I'll give you a simple example. Write down, all of you. For example, for your same structure is basically your simple tissues. Simple tissues. Simple dimple. Okay. Simple tissue. While your dissimilar are the complex tissues. Complex tissue. Didn't understand? Okay. I'll give you a better example. Listen very carefully. Under simple tissue, all of you know parenchyma. All of you know parenchyma. All of you know collenchyma. Collenchyma. And last one is what? What is the last one? The last one is sclerenchyma. Sclerenchyma. Under complex tissue, can anyone tell me, under complex tissue, we have two different types of tissue here. One is your water conduction, one is food conduction. Anyone? That is your xylem and phloem. Clear? Xylem as well as your phloem. Clear, all of you? Clear? So, a tissue is nothing but a group of cells. They can have a common origin, common function. Can they have common structure? Yes, in the case of simple tissue. Will they have dissimilar structure? Yes, in the case of complex tissues, xylem and phloem. Xylem and phloem. Xylem and phloem. Clear, all of you? Any doubt here? Any doubt here quickly? If there is no doubt, everyone quickly fire emoji in the chat, because I want the energy to be extremely high today. Right? Extremely high energy today. Let's go. Now, next thing I'll teach you is something called as types of tissues. Ready, all of you? Something called as your types of tissues. All of you? Types of tissues. Ready? Types of tissues. Very simple chapter, students. Very, very simple chapter. Just focus. You will score out of 4 in this chapter. Write down what I'm telling you. Write down what I'm telling you. You will get the notes in my Telegram channel. And write down everything. Trust me. Trust the process today. Trust the process. Sit with me today till the very end. And no one can take away a single mark from anatomy from you. That's my promise from all of you. Types of tissues. Now, students, now every single time you divide into types, right? There is a criteria. There has to be a criteria or some kind of basis on which you divide. For example, we are dividing the types of tissue based on the ability to divide. Based on the ability to divide. Based on the ability to divide. Because there are some tissues. There are some tissues which can divide. Nice, they'll divide, make money and be happy. There are some cells, some tissues which cannot divide. Now understand, friends, the tissues which can divide. The tissues which can divide are called as your meristematic tissue. Are called as what? Meristematic tissues. Meristematic tissues. Meristematic tissues. Now what about the tissues which cannot divide? They are like, life is very simple. I don't need to divide in my life. Very simple life I have. Tell me, students, which are the cells, which are tissues which cannot divide? The tissues which cannot divide are basically your permanent tissues. Permanent tissues. Permanent tissues don't need to divide. They have a permanent job. They don't need to find new job anymore. Nicely, happily they are in life. Okay. Now understand the difference. Understand the difference, all of you. Now, for example, you have meristematic tissue. They are mostly the living cells. They are mostly the living cells. Mostly the living cells. Living cells. Also called as your dividing cells. Also called as your dividing cells. Under meristem, we have something called as your primary meristem and secondary meristem. Primary meristem and secondary meristem. Okay. Now, all of you tell me in the chat. Who are the people who doesn't have to work? They are called as a permanent tissue. Why? Because they don't have to find new work. They have to work. They don't have to find new work basically. Okay. Permanent tissues are the tissues. Permanent tissues are the tissues which have a specific function. They have a specific function as well as specific structure. They have a very specific structure and specific function. They do not need to divide anymore. Do not need to divide anymore. All of you be like meristematic first of all. Learn, divide. Okay. Now, the simple next question is, what about permanent tissues? Are they living or are they dead? Are they living or are they dead? What is the answer? All of you, tell me. These T cells, here I told you they are living cells. Permanent tissues are made up of, they can be living or they can be dead. Both possibilities are there. Both possibilities are there. At the same time, your permanent tissues are two types, simple and complex. There are two types, simple permanent tissue as well as your complex permanent tissue. Simple permanent again, parenchyma, collenchyma, sclerenchyma. Complex again, xylem and phloem. Simple, done. One more topic done, students. If you understood this topic again, fire emoji in the chat because I want the entire chat to be running today. Full ultra level today, okay? Sir, which material can you prefer for biology? NCERT enough. NCERT is enough. Okay, don't worry. NCERT is enough. Okay. Now, students, we have simple tissues, complex tissues. Permanent tissues having all cells similar in structure and function and are called as simple tissue. Similar structure we spoke about. While your complex tissues are what? Different types of cells. We learned this. Different types of cells and they are called as your complex tissues, xylem and phloem. Xylem and phloem. Done, done, done. Now students, I want everyone to play a small game. Quickly, quickly, all of you. I want everyone to notice the diagram right here. This is a dividing cell. Spoiler alert. This is a dividing cell. It is a meristematic tissue. I want everyone to tell me the characteristics which you observe right here. All of you. Now, first of all, if you observe this diagram right here, what can you tell me? What can you tell me, students? Okay. Tell me the size first of all. Are they small or the large cells? First point will tell me, sir, they are dividing cells. I'll tell right down here. Whatever you observe here, you're supposed to tell me, I will write here. And all the students who are watching recorded also, pause, tell me and then read here. Okay. I will tell they are dividing cells. They are dividing cells. Okay, that I told already. Okay. Can you see there are small cells? Small cells. Small cells. Okay. They are small cells. Now students, do you notice anywhere intercellular space? All of you? Cell chapter you want to learn now. I know I can see cell division here. I can see probably metaphase here and anaphase here. I can see that also. But that is not the case. Try to tell me what is there about meristematic tissue. No, that chapter will tell later on. Done it already. Okay. Now do you notice anywhere intercellular space? Anyone? Do you see anywhere intercellular space? Anywhere? Obviously, intercellular space is completely absent here. Completely absent. Okay. Apart from that, do you notice they have dense cytoplasm because it's dividing cell? Dense cytoplasm. Dense cytoplasm. Okay. Dense cytoplasm. Okay. Other than that, what do you notice, students? Do you notice any cell wall here? There will be cell wall, dividing cell. Since it's a dividing cell, it will have something called as your primary cell wall. Primary cell wall. Obviously. Now, students, do you think the vacuoles will be filled here? Dividing cells, obviously vacuoles will be empty or vacuoles will be absent here, or sometimes depending upon the cell, vacuoles can be very large also. Okay, let me tell you based on that also. Okay. Now students, apart from that, do you notice nucleus? Do you notice nucleus everywhere? One bottu, it is there. Can you see? You might one round bottu, girls wear no one round bindi, one big one. It looks like that. No, see this. Do you notice one nucleus? You will see, yes sir. Do you notice the nucleus is very large here? Like, yes sir, very large nucleus. Okay. Do you notice the nucleus in center only? It is not in one corner. Obviously. So can I write? They have large central nucleus. Large central nucleus. So every single time you think of a meristematic tissue, this is what is supposed to come to your mind, and this is the points which you need to remember about meristematic tissue, also called dividing cells, small cells, intercellular space is absent, primary cell wall, dense cytoplasm, large central nucleus. Large central nucleus. Okay. Sir, Telugu, Abhaya. Okay, Abhaya only. See that only, Abhaya. Okay, see large nucleus, thin walls, no intercellular space and dense cytoplasm. If this following points are clear to all of you, quickly fire emoji in the chat because I want everyone to be active today. Keep learning, learn today, don't keep it as a backlog, understand in my class and just go home happily and sleep off. If you're in the house, sleep after the class, aram se. Okay, with satisfaction. And you learned something now. Okay, you learned something today. Now students, that was your meristematic tissue. That was your meristematic tissue types, base, broad, or meristematic tissue. Now students, can I classify meristematic tissue now? Now students, remember, every single time you want to classify the meristematic tissue, or matter of fact, you want to classify anything, there has to be some criteria. Yes or no? Yes or no? There has to be some criteria. For example, I am separating all of you into categories. Some of you are listening to me properly, making down notes, following what I'm saying. Some of you are listening, looking at the wall and nicely enjoying. There are two types of students. Every single time you want to divide something, there has to be a criteria. Right now also, if you want to divide the meristematic tissue, the criteria about dividing is two main categories. You can divide the meristematic tissue, students. Listen very carefully. you can divide the meristematic tissue based on two different criteria, that is based on origin and based on location. Based on the origin as well as based on the location. Two different criteria of dividing. Clear all of you? Clear? Two different criteria for dividing. Ready all of you now? Ready? Now first one we'll do is based on the origin. Ready all of you? First one we'll do is based on origin. Now based on origin, students, based on origin we have something called as, based on origin we have something called as your primary meristem and secondary meristem. We have something called as your primary meristem as well as, too lazy now, suddenly I became lazy, secondary meristem. Okay, now tell me students, in your school days, primary will come first or secondary will come first? Tell me all of you, your primary will come first or secondary will come first? You'll be like, obviously sir, I will go to primary class, then if I clear everything, if I don't fail in class 5th, they'll push me to secondary, then higher secondary, then you go to what is that, class 10th and 11th called as? See, I used to call it class 10th and 11th. There are different names, PUC or something, basically college you will go later on, right? So in your college, in your school life also, we had the primary then secondary. In the plant's life also, we have something called as your primary and secondary stage. In the primary stage, the meristem which is present is called as your primary meristem. It is present from the beginning. It is present from the beginning. It is present from the beginning. That is called as your primary meristem. Now, if it is present from the beginning, what is the main function of it? Obviously, it is responsible for the primary growth. It is responsible for the primary growth. Primary growth. Now, you will ask me, sir, what is the meaning of primary growth here? The meaning of primary growth is increase in height. Increase in height of the plant. Increase in the height of the plant, that is called as the primary growth. Clear all of you? Clear? Any doubt here? Now, this primary meristem is present in both monocot as well as dicot. Present in both monocot as well as your dicot. Done. Now, what is secondary meristem? Obviously, you go to secondary school later on. So, can I tell right now, secondary meristem is formed later in the life? It is formed later in the plant's life cycle. Later in the plant's life cycle. Okay, what is it responsible for? Here we learn it is responsible for the primary growth. Secondary meristem is responsible for the secondary growth. Here we will learn about two different concepts or two different structures, that is called as your vascular cambium as well as your cork cambium. Clear? Now students, understand, when I say secondary growth, what do I mean? The meaning of secondary growth is increase in width or lateral growth. Lateral growth, increase in width. Now understand all of you, the secondary meristem is only and only found in the case of your dicots. Done. Any doubt? First topic done. So you can classify the meristem. Based on the origin. When are they formed? Are they formed in the starting stage of plant's life or they formed in the later stage of plant's life? If they are present from the starting, it is called as primary meristem. Primary growth, height, present in both monocot and dicot. Done. if they are formed later in plant's life, if they are formed later in the plant's life, it will do secondary growth. Here there are two concepts called as vascular cambium and core cambium, mainly responsible. Secondary growth means increase in width or lateral growth, present only in the case of your dicots. Present only in the case of your dicots. Only dicots. That was based on origin. Anyone, any doubt, quickly ask me in the chat. If there is no doubt here right now, fire emoji in the chat so we can give the whole George going on. Okay, let's not drop the energy today. Okay, now next one is based on location. Next one is based on the location in which plant part we find meristem. Based on that, we are classifying now. We are classifying. Okay, understand, all of you, understand. Very simple to understand. Based on location. Based on the location. Based on the location, there are three different types actually, not one or two. Based on location, there are three different types here, students. Understand. Based on location, there are three different types. The first one is called as what? The first one is called as your apical meristem. Apical meristem. Now understand, students, understand what is apical meristem. What is apical meristem? Okay. Apical meristem is present. It is present at the growing root and shoot tips. Now, what is the meaning of apical? What is the meaning of apical? Apical means apex, top. Apical means apex. So, growing shoot tip or root tip. For example, understand. Very simple it is, very simple. If this is the soil right here. Here we have root. Here we have root. Lateral roots coming out. Here we have root. And we also have the shoot. We also have the shoot on top. Okay. In the growing tips, right, you can see right here in the growing shoot tip, and the growing root tip, that is root apical meristem and shoot apical meristem, we find the apical meristem. So meristem which is found in the shoot area, the meristem which is found in the shoot area is called as shoot apical meristem. The meristem which is found in the root, that is called as R for root apical meristem. Is that point clear to all of you? If someone asks you, where do you find apical meristem? We find apical meristem in the growing shoot tip and root tip. That is why it's called as what? SAM, Sam and Ram. Ram and Sham. Okay, Ram and Sham. So, Ram, Sham. Shoot apical meristem, root apical meristem. Root apical meristem. Now, students, one more extra point here. Understand, there is something called as axillary bud. How many of you know what is axillary bud or axillary meristem? You can call it whatever you want. Your apical meristem will also give rise to the axillary bud. Remember that. Axillary bud. Your shoot apical meristem will give rise to axillary bud. And your apical meristem is a type of your primary meristem. Now, why do I call it primary meristem, students? Why do I call it primary meristem? Because it is a meristem which is present from the young stage. It is a meristem which is responsible for the increase in height. So can I call the apical meristem a type of primary meristem? You're like, yes sir. Yes. Easy, done. Any doubt here? Quickly ask me, students. Quickly ask me. Now the second one. Second one is very funny. I don't know why I always laugh at this. It's called as intercalary meristem. Intercalary meristem. Intercalary meristem. Now, where do you find location? Location. See, we are classifying based on location, right? Classifying based on location, so you should write location here. It is present between. It is present between mature or permanent tissue. It is present between mature tissue. It is present between the mature tissues. Now understand all of you, very simple, I'll tell you exactly the application of it now. Now, how many of you like going on the grass and sleeping on the grass like this? Or if you play football or if you play some games, playing in the grass is so fun, right? Okay, imagine every single time you play in the grass or a cow is coming, a cow, C-O-W, cow, okay. A cow is coming and it is eating the grass. But the next few days, within next few days, the grass is growing again very fast. The grass is able to grow very fast. How? Because of the increase in the internodal region. Now, you'll ask me, sir, what is the internode? If this is the stem, this is a stem right here. This is nodal region. Nodal region. This is called as node, from which usually the leaf arises. Leaf arises at the nodal region. Okay. Between the two nodes is called as internode. Internodal region. Now, in the case of grasses, the internode is getting elongated very fast. Why? Because in the internodal region, or especially at the nodal region here, between the two mature cells, we have the intercalary meristem. We have what? We have the intercalary meristem, and because of this intercalary meristem, there is increase in what? Increase in internode elongation. Internode elongation. Now, which type of tissue is it? Is it primary tissue or primary meristem or secondary meristem? Your intercalary meristem is a type of your primary meristem. Primary meristem. Done? Done, students? Done? Any doubt here? Any doubt here, students? Any doubt here? Second one done? No doubt? Yes, cow. The thing is, for some reason, I've always noticed every single time I say an animal name, you will misinterpret. I'm like, what is this logic? Okay, the last one. Easy, right? Till here, any doubt? The last one is called, as all of you know, the lateral meristem. Lateral meristem. And students, remember, all of this is based on what? Based on location. Where it is present, I will show you. Okay, I'll show you. Now, where do you find lateral meristem? Where do you find lateral meristem, students? What is the meaning of lateral? Meaning of lateral means side. Lateral means side. So, it is present at the sides. What is the meaning of it? I will show you. Present at the sides. Present at the sides. For example, if this is a stem right here, nodal region, and this location right here, in this location right here in the sides, we will find the lateral meristem. We will find the lateral meristem. Lateral meristem in the sides like this. In the sides. Now here, as usual, always tell, here you will study something called as your vascular cambium and cork cambium. We will study about vascular and cork cambium. Okay. Now, unlike your apical meristem and intercalary meristem, your lateral meristem is primary in origin. It is secondary in origin. It is secondary meristem. It is a type of secondary meristem. Okay, secondary meristem. Function, all of you know. The function of lateral meristem is increase in girth. Increase in girth. Okay. Any doubt here, students? Any doubt here? We finished about what we finished till now? Till now we studied. I'll give a small recap for all of you who joined late. We started off with something called as plant tissues. What is tissue? Group of cells, common origin, common function. Will they have common structure? Yes, in the case of simple tissues. Dissimilar structure in the case of complex tissue. Done. Then we learned about type of tissues based on ability to divide, meristematic tissue, permanent tissue. Living cells, dividing cells. Permanent tissues, simple and complex, specific function, specific structure, living or dead. Then we learned about meristematic tissues, all the different characters. Then we started off with your meristematic tissues based on origin, based on location, both are done. Based on origin, we have the primary meristem, secondary meristem, present from the beginning. It is formed later on in the plant's life cycle. Primary growth, secondary growth, monocot and dicot, only in the case of dicot. Only in the case of dicot. Okay. Now, apical meristem, intercalary meristem, lateral meristem. Each one of them is based on the location. Based on the location. First one is called as apical meristem, present at the apex. Intercalary meristem present in the mature tissues, between the mature tissues. Lateral meristem present on the sides here. Okay, on the sides. And as usual, lateral meristem is only found in the case of dicots. Only in the case of your dicots. Till now, any doubt, all of you? Because if there is no doubt, I will ask you one difficult question in biology. Very difficult question in biology. Difficult question. Ready, all of you? Ready for a difficult question? All of you? Ready for a difficult question? Okay. The question is very simple. I'll give you 1 minute to think also. From primary meristem, do we get secondary meristem? Simple question. From primary meristem, do we get secondary meristem? Yes, sir. If thumbs up in the chat, if yes. If no, thumbs down in the chat. All of you, go. Go. I'll give you 30 seconds to think. Think. Use your brain cells now. Don't feel sleepy. Use your brain cells. Thumbs up if it is correct. Thumbs down if it is wrong. Students, this is what I tell in my class always. You can learn everything in the world, but if you don't learn the basics right, you will always end up losing some marks here and there. Like, you might have learned this chapter really well before. You might have learned about the entire concept of dicot stem, dicot root, monocot stem, dicot leaf, monocot leaf. But if you don't know the basics right, which unfortunately many students skip learning because they think it is very easy to learn. That is where you're going wrong. You can create a very large building, but the foundation is not good, right? The building will collapse. Today is the day you learn basics also. Never in the life cycle of plant, it will never happen that a primary meristem cannot convert it to secondary meristem directly. Primary meristem will never convert to secondary meristem. It will never convert. Now, a simple question. How many of you knew this? How many of you did not know this? Because you end up by hearting this part and it just stays like that. Just stays like that. Okay. So, what is the answer? It is not. Primary meristem does not give secondary meristem. Does not give secondary meristem. Now, are you curious? Then what is the answer? Are you curious now for the answer? Understand. I'll teach you again. We learned all this, right? We learned on this. Primary meristem, primary growth, increase in shoot length. Secondary growth, increase in girth. Lateral meristem, we learned all this. So students, remember, your primary meristem responsible for primary growth will always produce. A primary meristem responsible for primary growth will always produce primary tissues. Primary tissue. Xylem and phloem. Or it could be parenchyma, collenchyma, sclerenchyma. Primary tissues. Primary meristem will always produce primary tissues like your primary xylem, primary phloem. Primary xylem. Right? Primary phloem. Now this primary tissues, one example I'll give you, famous example which I always use. One of the few examples which I know, which is the best example, is medullary rays. Medullary rays, which is a permanent tissue, primary tissue, permanent tissue. Primary meristem will never convert to secondary meristem, but in fact, a primary meristem will give rise to a primary tissue. This primary tissue will be converted to secondary meristem. And the entire process is called as your dedifferentiation. It's called as what? Dedifferentiation. Okay? No. Okay? Dedifferentiation will give you secondary meristem. Now this secondary meristem will give you rise to secondary growth, will produce secondary tissue. How many of you understood? How many of you did not understand? Quickly ask me. Now, let me teach you one more concept as if I'm already teaching you. Right? Primary meristem to primary tissue. This stage is called as your differentiation. Secondary meristem, this stage right here, is called as redifferentiation. I will explain you only once. Listen to me very carefully. Only once I'll explain you. What is the dedifferentiation? What is differentiation? And what is redifferentiation? Understand very carefully today. Last time I'll teach you today. Okay. The meaning of differentiation. Is that, understand, meaning of differentiation is that a meristematic tissue is losing the ability to divide. Losing the ability to divide. This is a dividing cell right here. This dividing cell lost the ability to divide and it produces the primary tissue after losing the ability to divide. That is called as differentiation. Then what is the dedifferentiation? Dedifferentiation is nothing but it is regaining the ability to divide. Ability to divide. That is called as your dedifferentiation. A primary meristem will never make secondary meristem. A primary meristem will make primary tissue which undergoes dedifferentiation, regaining the ability to divide and form secondary meristem. And now this secondary meristem again it loses the ability to divide. That is called as your redifferentiation, which makes up the secondary tissue. How many of you understood? Now quickly raise your hands, fire emoji in the chat, do whatever you want in the chat now. Go. Understood? So students, this is the basic concept and many students skip this part. They keep skipping this part, I don't know why. But I want my students, my young botanists, to not skip the basics and understand always the flow chart. Done? Beautiful. Now students, remember I told you the location. I'll show you. Here is the location right here. See this. Apical meristem present at the apex. Intercalary meristem present at the nodal region. Lateral meristem present on the sides. Here. Done. Sir, how secondary meristem Meghana? Understand Meghana? You end up getting secondary meristem because of dedifferentiation. Your primary permanent tissue, for example, your medullary rays is regaining the ability to divide. This medullary rays, if it regaining the ability to divide, it is becoming vascular cambium later on. I'll teach you which vascular cambium I'll teach you now. Okay, clear? This primary tissue will regain the ability to divide and make up the secondary meristem. It is present in the starting stage of plant's life. It is present on the later stage in plant's life. Simple. Now students, can we decode this diagram, all of you? Can we decode this diagram given you NCERT? Very simple diagram, but let's decode the diagram one by one, point by point. Understand. Very simple it is. Very simple. Okay. Now first we will look at this diagram right here. Now tell me students, is this root or shoot? Tell me first of all, is this root or shoot? In the chat quickly go. This right here is a shoot. Can you see here? Right here we have the ram. Root, apical, meristem. We have the root apical meristem right here. Now this root apical meristem will divide on the both the sides here. Divide here and it will divide on the top. Divide on both the sides. When this root apical meristem, it is dividing on the lower side, it makes something called as initials of root cap. The meaning of initial is precursor or starting stage. So initials, like in your name also have initial, no, starting part. Okay, initials of root cap, and this initials of root cap will eventually develop into the actual root cap. Initials of root cap. On the top, do we have your initials of central cylinder and cortex? Okay. Now on the top, do we also have protoderm? Protoderm basically makes up the epidermis. It eventually develops into epidermis in the case of root, which is also called as epiblema. Point to be noted, epiblema. Initials of central cylinder will end up making the central cylinder. Now students, what is the central cylinder? If you know the structure of a root, in the center, do we have the vascular bundles? Yes. So the central cylinder is nothing but the vascular bundles in the center. Okay. Initials of central cylinder and cortex. So we are making the cortex also. So on the top, what do we have? We have the epidermis, cortex and stele. Epidermis on the outside, cortex the middle layer, and the vascular bundles on the inner side. Done? Decoding done? Students? Very easy to understand. This chapter is very easy if you follow the following points. Okay. Now, let's go to the next point here, all of you. Let's go to the shoot right now. Shoot. Now, what do we have here? Can you see a meristematic tissue? A primary meristematic tissue here. Right here. This right here is your shoot apical meristem. SAM. Sam uncle. Okay, Uncle Sam. Sam here. This SAM will divide on the both the sides. Yes, divide on the both the sides. When it is dividing on the bottom side, it makes up the differentiating vascular tissues. Different types of conducting tissues are made. Okay. On the top we have the leaf primordia, which will develop into the leaf. Now students, understand, if you know a little bit of morphology chapter also. If you know morphology chapter, on the top, students, on the top of a stem, we have some kind of bud. Can anyone tell me which bud do you find on the top? Anyone in the chat? This is stem. These are the nodal regions. On the top, we find something called as your apical bud. How many of you remember? Apical bud on the top. Now I have always told you, if the apical bud is growing, on the top, as the apical bud or the shoot apical meristem grows, leaves are produced in an acropetal succession. As the apical bud is growing, leaves are producing in an acropetal succession. What is the meaning of acropetal succession? Older leaves at the bottom, younger leaves at the top. Acropetal succession. So as the apical bud is growing, Now students, understand, this is a very important line of NCERT right now. As the apical bud is growing, as the shoot apical meristem is growing, as it is growing on top, some cells are left behind. In the leaf axis, this is called as leaf axis. This is called as your leaf axis. In the leaf axis, some cells are left behind there. Can you see here? In the leaf axis, some cells are left behind, and these cells make up something called as your axillary bud. They make up the axillary bud. Now do you see it here? Can you see here? In the leaf axil, the shoot apical meristem is growing. Some cells are left behind, which makes up the axillary bud. And this axillary bud right here, it can be converted to, it can grow into a branch or a flower. It can develop into a branch or a flower in the future. Students, isn't botany very easy? Tell me now. Isn't botany very easy when you visualize and learn? When you decode the NCERT, when you learn the diagrams properly. Botany is very, very easy, students. There's nothing to by heart here. Nothing to by heart. Understand what is then given diagram. Everything is easy. Okay. Beautiful. Now can we read the lines of NCERT? Tell me, students, can we read the lines of NCERT now? I want some energy in the chat, all of you. I feel you're already going down. Already going down. Show some energy in the chat, all of you. Now, we will read the lines of NCERT. You will tell me, sir, NCERT is easy. NCERT is like so easy, I can read with my eyes closed also now. See this now. Growth in plants is largely restricted to specialized regions of active cell division called as meristems. So meristems are the regions where active cell division will take place. Yes, sir. Okay. Plants have different kinds of meristem. The meristem which occur at the tip of the root and shoot and produce primary tissue. So primary tissue present at the apex, shoot tip and root tip. What do you call them? Apical meristem. Apical meristem, which is a type of your primary meristem. Done. Now you read. Root apical meristem occurs at the tip of the root. Obviously, sir. While the shoot apical meristem occupies a distinct most region of the stem apex. Obviously, during the formation of leaf, elongation of stem, as the SAM is growing on top, as the shoot apical meristem is growing, some cells are left behind from the shoot apical meristem. When your Uncle Sam is growing on the top, as the shoot apical meristem is growing, some cells are left behind, and those cells constitute the axillary bud. Such buds are present in the axis of the leaf, in the axil of the leaf. I showed you which is axil of the leaf, and are capable of forming a branch or a flower. Done. Such an easier paragraph to understand now. The meristem which occurs between the mature tissues or permanent tissue is called as your intercalary meristem. They occur in the grasses and regenerate the part removed by the grazing herbivores, the cow. The cow is coming and eating and done. Okay, cow is coming and eating and internode is elongating. Both apical meristem and intercalary meristem are called as your primary meristem. Primary meristem, done. Because they appear, see, why are they called primary meristem? Because they appear early in the life of the plant and contribute to the formation of the primary plant body. That is increase in the height. Increase in the height. Done. Any doubt in this paragraph, all of you? Done. Now, students, as usual, always, every single time I teach this chapter, I find that mistake all the time. Okay, there is a small mistake here. But it is not technically a mistake, but grammatically it is not well explained here for students to understand. I'll tell you first what is it, then I will explain. Understand this. The meristem that occurs in the mature regions of the root and shoot of many plants, particularly those produce woody axis and appear later in the plants, later in the primary meristem, is called as secondary meristem or lateral meristem. Now it says or here. It says or here. So will any child assume secondary meristem and lateral meristem are same to same? Will you assume secondary meristem and lateral meristem is same to same at this point? Like I said, it says or. English or means same to same, synonym type. Okay. Or lateral meristem. Okay, understand, I'll tell you the difference now. They are cylindrical meristems. First one, that is called as your fascicular vascular cambium. First one, interfascicular cambium and cork cambium are examples of lateral meristem. They are examples of what? PYQ alert. PYQ. They are examples of lateral meristem. Now, students, all of you will think right now. The NCERT line is telling you fascicular vascular cambium, interfascicular cambium, and cork cambium. Three different cambium is telling you they are examples of a lateral meristem. In the previous slide, you learned that secondary meristem and lateral meristem are same to same. Can you draw conclusion from the above line and the below line? Any one conclusion you can draw. Your core cambium, fascicular cambium and interfascicular cambium, all three of them are not only just lateral meristem, but they are also secondary meristem. Yes or no? Tell me, all of you. Axillary buds are derived from the activity of SAM. Activity of SAM. Shoot apical meristem. Tell me, yes or no, all of you. You will think that your fascicular vascular cambium, interfascicular cambium and core cambium, since they are example of your lateral meristem, they are also the example of secondary meristem. Yes or no? Tell me, will you think like that? Yes. And this is where many students can go wrong. Many students can go wrong here. Let me tell you why. I will teach you a little bit extra information, like I'll show you the diagram representation. Then you understand the definition of this line here. See this, all of you. I will tell you exactly where we find each one of them. All of you pay attention now. All of you pay attention here. All of you pay attention here. Okay. This is a root. This is a section of a root or a stem. Okay, section here. Okay. In this section, we find vascular bundles here. Okay, we have vascular bundles here. Okay. Xylem, phloem. Xylem, phloem. Xylem, phloem. Xylem, phloem. Xylem and phloem. Now understand, students. Between xylem and phloem, there is a cambium. Okay. Between xylem and phloem, there is a cambium. In between the vascular bundle, there is also a cambium. Okay. Understand now, which is what? The first of all, let's focus on the green color one. The green color one, okay? The green color one. The green color one is called as your fascicular vascular cambium. Fascicular vascular cambium. It is also called as your intrafascicular cambium. Intrafascicular cambium. Intrafascicular cambium. Intra, within. Intra means within. Okay. Now focus on the red one. The red one here. The red one is called as your interfascicular cambium. This one is interfascicular cambium. Interfascicular cambium. Okay. Understand, all of you, understand. Your interfascicular cambium, students, very important line of NCERT right now. You will thank me with this. The green color part is within, right? It is within the vascular bundle, while your interfascicular cambium is between the vascular bundles. It is between the vascular bundles, inter. Now here, students, very important line of NCERT, not given NCERT, but let me tell you, your fascicular vascular cambium, which is present between xylem and phloem, it is actually a type of primary meristem. Okay, within. While your interfascicular cambium, which is present between xylem and phloem, it is your secondary meristem. So please remember, students, please remember this throughout your life. In this region right here, in this region right here, we had something called as medullary race. Medullary race. We learned about this somewhere here. We learned about medullary race here. Medullary race, permanent tissue. This permanent tissue underwent dedifferentiation. It underwent dedifferentiation to make the secondary meristem. So this particular structure right here, that is your medullary race, underwent what? Underwent dedifferentiation. Dedifferentiation to make the secondary meristem. To make up the secondary meristem. I hope this is very easy to understand for all of you. Very important lines of NCERT. Many students go wrong here. So, but understand. Okay. Fascicular vascular cambium or intra means what? Within primary in origin, because nothing was here before. This tissue right here did not undergo dedifferentiation or differentiation yet. It never went under dedifferentiation or just differentiation also. Normal primary meristem. While the tissue which is present here, that is interfascicular cambium, it was medullary race here first. Those medullary race underwent dedifferentiation to make the interfascicular cambium. Okay. So this, I hope all of you are able to understand botany now. Don't by heart any of this. Okay. So primary in origin, secondary in origin, and secondary in origin. But all three are examples of what? All three are examples of your lateral meristem. Lateral meristem. What is the main function? Producing secondary tissues here, most of them. Okay. I hope all of you understood this. Now, following the division of the cells in both primary as well as secondary meristem, the newly formed cells become structurally and functionally specialized. Differentiation. Okay. Such cells are called as your permanent or mature cells. Constitute the permanent tissues. During the formation of primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. That is basically differentiation. They're talking about your differentiation. Okay, differentiation. All of you know this? All of you know this? Yes or no? Lateral meristem, how do we have? We have interfascicular cambium as well as core cambium. Your intra is also called as what? Fascicular vascular cambium. Same meaning. Same meaning. Okay. Shoot. Your NCERT. Question from NEET 2022. Time starts now. Quickly go. You'll have only 10 seconds. You have 20 seconds. Go take 20 seconds. Enjoy 20 seconds now. And students, all of you are asking me the syllabus for this chapter, right? So all of you remember, students, secondary growth is removed. Secondary growth has been removed, but the tissues part is still there. Tissues part is still there. You have to study the tissues from the old NCERT. That is why we are doing it here. That is why we are doing it here. Okay. Axillary buds are derived from the activity of. Did we learn here? Axillary buds are derived from the activity of. Axillary buds are derived from the activity of. I told you, as the apical bud or the shoot apical meristem is growing on top, axillary buds are forming here. Yes or no? Yes or no? What is the answer? Apical meristem. Apical meristem, not lateral meristem. Apical meristem. So as the apical meristem is growing, the top one, as the apical meristem is growing, axillary bud is produced in the leaf axil. Okay. Minus for all of you who told wrong answer. Okay. Just copy paste, huh? Cut. Understood? Students, I'll tell you again. Apical meristem. Here. I think I wrote also here. Can you see this? Can you see this? Apical meristem will also give you what? Axillary bud. Also give you axillary bud. Brain cells, no? Tuk. Oh God. Okay, one more question. Let's see. One more question. Okay. Consider the following plant tissue. Identify the lateral meristem. Identify the lateral meristem among the following? Okay, identify the lateral meristem. Axillary bud? No. Fascicular vascular cambium? Yes. Interfascicular cambium? Yes. Cork cambium? Yes. Intercalary meristem? No. So what is the answer? B, C and D. B, C and D is here. Next question. Okay. Interfascicular cambium is present between. Interfascicular cambium, not intra. Interfascicular cambium is present between primary xylem and primary phloem? No. Pericycle and endodermis? No. Two vascular bundles? I'll tell you. Second xylem, second phloem? No. Between the two vascular bundles? Right answer. Here, we learned here, right? Here. Interfascicular cambium is very present. It is present between two vascular bundles. Vascular bundle number 1, upper 2. Between the two, we have the interfascicular cambium. Interfascicular cambium. My God. Now, students, with that being said, now students, can we start with something called as permanent tissue? Can we start with permanent tissue now? Can we start? Yes, sir. My water is also over. Now we will start with something called as permanent tissue. Okay. Now students, understand very simple is permanent tissues are also divided into two categories. Permanent tissues are also divided into two different categories. Understand, very simple it is. We have something called as your simple permanent. Simple permanent. Simple permanent and complex permanent. And all friends, quickly smash that like button, all of you. Quickly smash that like button and I'll teach you what is this. Does the parenchyma have we'll come to parenchyma now. We'll come. Okay. Now, what is the difference here? What is the difference between simple and complex? Very simple definition. Understand the simple definition and you will don't need to even break your head. Your simple permanent, it will have only one type of cell. Here, more than one type of cell. More than one type of cell. That's all. Done. That's all the difference. That is, simple permanent will have only one type of cell. Example is going to be what? Example is going to be your parenchyma, collenchyma, and sclerenchyma. Here we will have the xylem and phloem. And the phloem. Xylem and the phloem. Xylem and the phloem. Xylem and the phloem. Such an easy thing. Friends. Now, we will not be focusing on the simple permanent tissues. Simple permanent tissues. Tell me, students, how many of you face difficulties in remembering this? Like, do you face difficulty in remembering this entire thing? Sir, what about parenchyma, sclerenchyma, collenchyma? I keep reading this, but I keep forgetting it. Understand, students? We will learn in today's class what is given in the diagram and then write it down. Here and there, I'll tell you a few extra points. You don't have to study anything outside of it. Okay. For example, under simple permanent, we have the parenchyma, collenchyma, sclerenchyma. Right. Now, first of all, tell me, what do you see in the diagram? And then you tell me, I'll write it. All of you. Tell me. It's easy only, I know, but see the diagram and tell me, all of you. See the diagram and tell me. Okay. First of all, can I tell? You keep hearing always parenchyma, 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 parenchyma all the time. So first of all, can I tell? It is the most common tissue. Most common tissue. Most common tissue. Okay, done. It is the most common tissue. Now, what is the function of it? The main function of parenchyma is packaging. Okay, main function is packaging. Like, every single time you see one empty space, no, stuff with parenchyma. Every single time you see empty space in the case of plant, you stuff with parenchyma. Keep stuffing parenchyma there. So main function is what? Main function is packaging. Okay, main function is packaging. Okay. Can I also tell it is the major component in all organs? Major component in all organs. You keep hearing parenchyma everywhere. Okay. Now, anyone look at the shape of the cell. Can I tell, if you look at the shape of the cell, iso means what? Iso means same. Same diameter throughout. Can you see? Same diameter throughout. Okay. So can I tell the next point, which is from here only I can tell you. Can I tell cells are isodiametric? Cells are isodiametric. Okay, isodiametric cells. Cells are isodiametric. They can also be oval, spherical, all that is there, but mainly it's isodiametric. They can be oval, spherical, round, polygonal, elongated, all that is there. Okay, that is extra. Now, all of you tell me, I'll write out, otherwise you'll tell me, sir, you don't teach only. You can have various shapes. Various shapes are also there. Oval, spherical, round, polygonal, and elongated. There are many different shapes also are there. Varied shape. Now, students, do you see intercellular space anywhere? Do you see anywhere intercellular space? Anyone? Rope, exactly, rope. Oh, DNA students are telling me now. Rope, exactly. R-O-P-E. Round, oval, polygonal, elongated. Apart from that, you can also add the spherical also. Ropes. Okay. Round, oval, polygonal, elongated, as well as spherical. Ropes. Now, students, look at the intercellular space. Now, intercellular space is very controversial here because in some of the tissues there is very less intercellular space, or mostly absent in many cases. Very less intercellular space, or it is completely absent here. Okay. Now, apart from that, cell wall, they have very thin cell wall. And the thin cell wall is made up of what? It has cellulose depositions. That's all, nothing, over. Khel khatam. Okay, khatam. Now, understand all of you. Function, you understand. Function. Tell me, friends, what is the main function? What could be the main function of your parenchyma? Main function. I told you main function is packaging. So can I tell storage? Is the main function? Yes. Storage is the main function. Now, what if a particular parenchyma becomes photosynthetic? It becomes green color. So what is ICS? Intercellular space. Green color becomes photosynthetic. Yes, it can become. In such case, parenchyma is called as what? Parenchyma is called as chlorenchyma. It is called as what? Chlorenchyma. Okay, done. Now, what if your function becomes gas exchange? It can help in gas exchange. that time the parenchyma is called as what? Palisade parenchyma. Palisade parenchyma. Palisade parenchyma. Okay, palisade parenchyma, or it is basically spongy parenchyma. You might have noticed in the, you will notice in the, this thing, leaf, spongy parenchyma will be there. Spongy parenchyma will be there. Okay. Now, students, what if it can maybe I'll write here. I'll write here. What if it provides buoyancy? What if it provides buoyancy? Buoyancy means floating on top. Buoyancy means floating. Buoyancy means floating on top. In such cases, it is called as aerenchyma. It is called as aerenchyma. See, one name, multiple function. One name, multiple function. At the same time, it can also produce something. It becomes secretory. It can also becomes secretory in nature. Means it can start producing some things. Start producing some kind of solvent. Okay, solute or some kind of liquid. Also, okay. Done. 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 Students, I will recap one more time. Listen to me, all of you. I'll recap one more time for all of you. Nothing is difficult. First of all, it is a common tissue. Yes. Packaging. Yes. Major component of in all organs. Yes. Cells are isodiametric. Done. Various shapes. Ropes. That is round, oval, polygonal, elongated, spherical. Very less intercellular space or completely absent thin wall cells made up of cellulose. Function, storage, photosynthetic, gas exchange, buoyancy, and secretory. Done. Done. That's all. That's all is parenchyma, students. That's all is parenchyma. Nothing more than this. Nothing more than this. Done. Any doubt in this paragraph? Quickly ask me, students. Any doubt in this paragraph? Quickly ask me, any one of you. Done. Parenchyma. That's all. Quickly fire emoji in the chat, all of you. Now, students, how many of you have? This is called a celery, or you can take your some of the cucumber, or not cucumber. This thing. What is that? Coriander also. Okay, coriander also, or cabbage. If you break it, do you find Meghana, examples of buoyancy? Pistia and icornia. You might have noticed, no? Pistia and icornia, right? Pistia and icornia. The water hyacinth also. It will float on water, nicely float on water. That is called as aerenchyma. Okay. Now, have you noticed this coriander? When you break the coriander, if you take a bunch of coriander and break it, you will hear that sound, something kruk 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 sound will come. Have you noticed that? When you break a celery also, some coriander, you break the stem, one kruk kruk sound will come. I'm not able to make the sound properly, but do you hear that some weird sound? Crunch crunch sound, crunch crunch, crunch crunch. Now, why the sound could be? From what is the main reason for that sound? Anyone? What could be the main reason for that sound? Is there some tissue? Is there some tissue which is breaking here? Huh? Is there some tissue which is breaking? Some tissue which can become photosynthetic also? Some tissue which can provide mechanical strength? It is a living tissue. We didn't write here, huh? Living tissue. It is a living tissue. Then you will tell me, sir, parenchyma is non-living. What way will I go then? Living tissue. Okay, it is a living tissue. Remember that. Which is the tissue, students? Which is the tissue? Which is the living tissue? Which can provide mechanical strength. Like there's no tomorrow. Nice mechanical strength will provide. It is also living tissue. It can also do photosynthesis. Exactly. Collenchyma. It is all because of collenchyma. The same collenchyma which is present in the stem, in the dicots. It's the same collenchyma. Same collenchyma. Okay. Same collenchyma. Okay. So what is collenchyma, students? Collenchyma is a living. It is a living mechanical tissue. Living mechanical tissue. living mechanical tissue. But in the case of colon-chyma, students, in the case of colon-chyma, you will never see intercellular spaces. You will never find intercellular spaces, completely absent here. Why? Because the cell wall has pectin depositions. Pectin deposits. Nicely pectin deposits are there. In the corner, lignified, complete, not lignified, pectin depositions. Okay, complete pectin depositions are there. Pectin depositions. The next question is, where do you find them? Where do you find colon-chyma? Where do you find colon-chyma? Do you find colon-chyma in the roots? No. Let me tell you, students, very important line of NCERT, not NCERT. Anywhere you will find this. First of all, it is completely absent. Colon-chyma is absent in monocot and also absent in roots. It is completely absent in roots. Okay, absent in roots. Roots of dicot. So then, if it is absent in dicot, it is absent in monocot. Where do you find it then? Where do you find it? Where do you think you'll find it? You will find this in the stem region and the leaf region of the dicots. Only stem and petiole mostly. Stem and petiole or the leaf part of the dicot. Otherwise, you will never find them anywhere. Okay. But exactly, if you look at the structure, if you look at the structure of your sectioning of a stem, upper part is called as your epidermis. Epidermis. The below the epidermis is called as what? It is called as your hypodermis. Hypodermis. In this hypodermis region, you will find colon-chyma. So where is it present? Present below the epidermis. It is present below the epidermis. Epidermis. Now students, what is the main function here? The main function is mechanical strength, obviously. The main function here is going to be mechanical strength. Before we go to the function, let me write down one more thing. Large vacuoles. Large vacuoles will be there. Okay. And also, some of them are living, so contains chloroplast. It contains chloroplast. Now tell me, students, what is the function here? Function. What is the main function here? The main function is obviously mechanical strength. The main function is going to be mechanical strength. Main function is going to be mechanical strength. Also photosynthesis, because it has chlorophyll. Photosynthesis. Okay, photosynthesis. Last thing is provides elasticity. Now, what is the meaning of elasticity? You can take the petiole. Can you bend the petiole? Yes, sir. The petiole is fluttering in the wind, nicely fluttering in the wind. Petiole. Why? Because of colon-chyma. You can take the stem, green stem, herbaceous stem, bend the stem will bend. Why? Because of colon-chyma. Young mint, jasmine, right? Mint plants, mint, you know, mint, biryani you add, no, biryani you add, right? That is bending of mint is because of your colon-chyma only. Okay. So elasticity. I will recap one more time. Living mechanical tissue, intercellular space is absent, cell wall has pectin deposits, present below the epidermis in the hypodermis region, large vacuoles containing chloroplast, mechanical strength, photosynthesis, elasticity. Three main functions of colon-chyma. Absent in monocot, absent in root of dicot. Then where do you find it? You will find only in the stem as well as leaf, especially petiole part. Now, a question to all of you. Simple question, students, simple question. Tell me, living plants also have dead cells? Yes or no? Question mark, question mark. Like for example, we are living, but we also have something called as your hair is there, nails are there, they are dead tissues. do you think even in the case of plants, there are some kind of dead tissues? Yes or no? Or everything is living? Entire plant, top to bottom, living only? Tell me, students. Is it top to bottom living, or are there any small dead tissues also present in the case of plants? Yes or no? Do they have dead tissues, or it's a simple question? Simple question. Do you think there are dead tissues here? Yes. The dead tissue which I am talking about is nothing but the sclerenchyma. The dead tissue which I am talking about is there is the sclerenchyma. Sclerenchyma is the simple dead tissue. It is a simple dead tissue. Simple dead tissue. That is without protoplast. Without protoplast. Without protoplast. Simple dead tissue without protoplast. Then what could be the main function here? Main function is only mechanical strength. Main function is mechanical strength here. Okay, mechanical strength. Now the cells are long, and the cell wall has lignin deposition here. Lignin deposits. Students, understand. In the case of parenchyma, cellulose. In the case of collenchyma, it was your pectin. In the case of sclerenchyma, it was your lignin. Lignin deposits. So remember all three of them. Many questions have come from anywhere. They can ask you for lignin, they can ask you for your cellulose, or they can ask you for pectin. All the three you need to remember at any cost. Remember at any cost. Okay. Then no. Okay. Now students, function. Before we go to the function, there is a classification here again. Now students, tell me. Whenever, every single time we classify, can we classify without a criteria? No. We need a criteria to classify. We need a specific reason to classify. And the reason or the criteria here is function, origin and structure. The criteria here is based on function, origin and structure. Based on function, origin and structure, the entire sclerenchyma is divided into two different categories. It is divided into two different categories, that is called as your fibers and sclerates. Fibers and sclerates. Two different things. Now fibers, what? First of all, tell me students, if you look at the structure, can you tell the difference? It is much more elongated with tapering ends, while this one is much more variable shape. It is round, oval, spherical, some goal-model thing this is. Okay. Can you see that there is a difference, simple difference in the entire structure also here? Like, yes sir, very different structure. That is thick-walled, elongated cells here. Generally occurring groups, they occur in groups here. Cell wall has pits. I'll tell you what is pits. Okay, they can have simple pit or bordered pits here. Simple pit or bordered pits. At the same time, sclerates, highly thickened cell wall, have narrow cavity. This is the cavity or the lumen. Very narrow. Narrow means very tiny, very small. Okay. Now students, I'll tell you what is a pit now. Understand what is a pit. For example, this is the plasma membrane. Outside the plasma membrane, we have what? We have the cell wall. In this entire structure, there can be certain depositions. Can you see? There can be certain depositions here. There are certain depositions. No. There are depositions in the cell wall. Deposits in the cell wall. But students, in this entire depositions, do you see some gaps? Do you see some gaps here? It could be here or it could be here. The gaps. These gaps are called as your pits. Okay. So there can be gaps in the cell wall. And those gaps are called as what? Gaps are called as pits. Gaps are called as your pits here. Okay. Now, where will the question come from? Many times the question have come from the location. Location. Okay. For example, your sclerets are found where? Found in the fruit walls of nuts, fruit pulp of guava and pear, and seed coat of legumes and tea leaves. That is the location. So in the examination, they will always ask you this part here, the location of sclerets. So in examination, if they give you dash following type of simple tissue is found in the fruit walls of nuts, fruit pulps of guava, legumes, tea leaf, what will be your answer? Tell me students, what will be your answer? Your answer should be what? Sclerets, not sclerenchyma. Proper answer will be, technical answer should be sclerets that time, sclerets. Okay, clear all of you? Sclerets is the answer. Now can you see the lumen? Lumen is completely filled here. Lumen is completely filled with the lignin deposits here also. Okay, call it obliterated lumen. See here, in the coconut we will have what? Sclerets will be on top. In the pear also, can you see when you eat this pear? When you eat the pear, do you notice that students, when in the mouth something will come? Or when you are eating chiku, you will drink chiku juice, no? I don't know what you call chiku in Hindi and other languages now. The chiku fruit. When you eat the chiku fruit, do you get that fibers? Do you get that fibers in your mouth? Sclerets only. In this walnut, extra outer structure, that is also sclerets. So all these following places we find sclerets. Okay. See here. Diagram is very important, students. Diagram is very important. NCERT diagrams. This is going to be a parenchyma. See, it's given here. Can you see here? Interstellar space is given in parenchyma. Given. Okay, this is your collenchyma. Lignin deposits. Can you see? Thickened corners because of the lignin depositions. Okay. Chiku is a fruit. Sapota. You know sapota. Juice you make. Sapota juice. Right. Sapota juice. I think I'm going colorblind. You're typing green and blue hearts are coming. Okay. This is your fibers, sclerets, pits. Can you see pits? Lumen is the middle part. Lumen. Thick cell wall. Okay. Students, that is the end of simple permanent tissues. Now students, can we start with the complex permutation now? Peach are different. Sapota, yes, sapota all of you know. You know sapota, right? Juice which you make. Sapota, yes, sapota you know. Exactly, Sakanya. The chiku juice. Okay, sapota juice you make now. I don't know what to tell now anymore. Okay. So when you're eating sapota juice, do you get something in your mouth? Like this fibers you'll get? No. That is part of your sclerenchyma only. Sclerenchyma. Okay. Students, ready for one fact? All of you ready for a simple fact? Okay. When you eat bananas, when you're peeling a banana, do you find that small thread-like structure there which you remove and throw off? When you're eating banana, do you get that small thread-like structure which you remove and throw off and like chi, I will only eat the banana, right? What do you think that is? Support ice cream is there, I know, from naturals. What do you think that is, students? That is actually the phloem fibers. That is actually the phloem thing, phloem. And that is actually helping in transportation. So don't throw it out next time, eat that also because it has a lot of nutrients. Don't throw it out. Okay, that is phloem actually. Okay. Now students, complex tissue we have something called as xylem and phloem. We have xylem and you will do, I know, 100% you will do. Okay, mango also, nicely you will remove the mango covering, eat only the inside part. Eat the entire mango, Tell me students, I'll ask you one more question now. Okay. In mango which you eat the outer part which you cut and throw, no? What is that called as? Is it your, which coat is it of a fruit? See, you eat miso, you eat the middle part, right? What is the outer part called as? If you know the chapter of morphology, you will tell me this answer. Okay. Now students, xylem and phloem. We have primary xylem. Obviously, primary xylem is first formed. Primary xylem first formed. Okay. Secondary is called secondary xylem, formed later on. Secondary growth. Secondary growth. Okay. That is also, surely that is very sad. Yes, chemicals are there. Okay. Eat everything. What? Oh no, no animal. You are animal only. Everyone. Epicarp. Oh God. Okay. Now phloem also we have what? Phloem also we have primary and secondary. Primary and secondary. Okay. Epicarp. Exactly. Epicarp, mesocarp, and endocarp. Now students, xylem. Now I will tell you one simple, ask you a simple question, very heavy question. Ready? Ready for a very, very heavy question, a very difficult question. Ready all of you? The heavy question is, okay, I am pro, you need to understand morphology chapter for it. Okay, morphology chapter. Okay. Heavy question, okay. Tell me the function of xylem, go. Let's see who tells the answer first. Function of a xylem. Quickly. All of you know, xylem is basically conducting tissue. It's a type of your conducting tissue. Type of your conducting tissue. Okay. Conducting tissue. Your xylem can be living or non-living. Both. It can be living or non-living in nature. Living and non-living, both possibilities are there. Both possibilities are there. Function, all of you know, conducts water and minerals. Water and minerals is the main function. Okay. Now students, tell me, okay, from here, this is the roots. The water is sent to the top. Okay. Is there a case where from the top water is sent down? Do we anytime get that? So all of you know, right? Water is sent from here to here. But do you send water from the top to bottom? It will never happen. It will never happen. It will never happen at all. It will never happen at all. So can I call it as the unidirectional flow? Unidirectional flow. Directional flow. Also provides mechanical strength to some extent. To some extent. Unidirectional flow. Okay. Now students, moment of truth. How many of you get confused here? Tell me students. How many of you get confused here between tracheids, vessels, xylem fiber and xylem phloem? It is very easy. I have prepared these notes for all of you. All of these notes will get on Telegram channel. All of you know, right? All of these notes will get on my Telegram channel. Because we are making amazing notes right now. All of these notes will be available on Telegram channel. Let me tell you that. It will be available on Telegram channel. Now, the game is very simple. You're supposed to tell me how many of the following are living, how many of the following are dead. Time starts now. Go. It's very simple, students. Very, very simple. Just listen to me. See the notes. You will fall in love with learning of it. Very simple. It is very simple. Okay. Tracheids, vessels, xylem parenchyma, xylem fibers. Now, tracheids are basically non-living. Vessels are non-living. Xylem fibers are non-living, but your xylem parenchyma is living. Xylem parenchyma is living. So can I tell there is a 3:1 ratio? 3:1 ratio. 3 being non-living and 1 being living. 3:1 ratio we are maintaining. Yes, 3:1 ratio we are maintaining. Okay. Non-living, living, non-living. Okay. 3:1 ratio or 1:3 ratio. Okay. Now students, all of you have tap in your house. Tell me, all of you have tap in your house. Tap is there. Water is coming in your house. Like, yes sir, I have water. Water is coming. In summer, water will not come. That is a different story. Okay. Now what happens is, think of your tracheids as small tap. Think of tracheids as a small tap. Tap you open or tap. Okay. Now the tap which is connected to your house, does it have a small pipe or a very large pipe? Tell me, students. See, this is how you remember it. Very easy to remember. Difference between tracheids and vessels, you'll never get confused. Okay. A single small pipe is coming. In a single small pipe, you're opening the tap, water is coming to you. Small tap is there. Okay. Now think of your tracheids as the small tap or a small pipe through which water is coming through to your house as tracheids. Just like how in your house water is coming from a small pipe, here also we have a small elongated pipe. We have a small elongated pipe with a tapering end. Like how in the pipe we have pipe and we have a tap, right? We have a tap here, right? Similarly, here also how we have a tapering end. We have a tapering end here, right? So here also we have tapering ends. We have the tapering ends. Okay. Small pipe with tapering ends. Okay. So elongated tube-like cells like a pipe with tapering ends. Thick lignified walls, because obviously if the water is coming, the wall should be very thick. If the pipe is not very thick, some local 10 rupees pipe if you add, it will burst and will open. No water in your house that time. Okay. So you should have thick lignified walls. Inner layers of the cell wall have lignin that vary in the form. So students, tell me, if there is so much lignin deposition here, lignified walls, will the poor pipe survive? Okay, like no sir, it will not survive at all. Dead it will become. So dead without protoplasm, because it is completely thick and completely covering is there, dead cells. One of the main, very important line, very important line right here. Very important line. One of the main water transporting elements of the xylem. One of the main elements of the xylem. Okay. So if you tell me water conduction is happening, it is mainly because of tracheids, not vessels. Okay. Main is tracheids always. 10 rupees pipe. Yes. Now students, now always remember your tracheids, like how we have only one pipe here, it is unicellular also. Unicellular in nature. That is all. Done. That's all is tracheids. Long elongated tube-like cells with tapering end. Okay. Lignified walls. Since it is very lignified on the inside also, without any protoplast, it is dead cells. One of the main water transporting elements of xylem. That's all. Understood, all of you? Understood? Yes or no? Understood? That's all is tracheids, student. That is all is tracheids. And tracheids can be found in your gymnosperms as well, also in pteridophytes also. Even in angiosperms it is found, but I'm telling you extra. Pteridophytes. Found in pteridophytes. Difficult, tell me. Difficult? Any difficulty here? Obviously not. Absolutely not. Now, think of vessels as a large pipe. Okay, big pipes are there. Have you noticed? From bathroom, one big pipe will be going to the gutter. From bathroom, one big pipe will be going to gutter. You would have seen. And that big pipe will have multiple pipes connected to it. From one room, one pipe is coming. From neighbor's room, one more pipe is coming. All the pipes are coming and attaching. Everything is going to gutter later on. Transport of water is happening with waste. That is different story, but transport of water is also happening. Yes. Okay. Now, vessels are like your big pipes. Okay. And which can stack on one another. Can you see here? They can stack. So, this is one vessel. One more vessel will be here like this. One more vessel will be on top. Can I call them as multicellular now? Multicellular. They're multicellular. Okay. Long cylindrical tube. Think, students, trust me, just close your eyes. One nice gutter ka big pipe will come. Gutter's big pipe is coming to your mind when you think of vessels. Large cylindrical tube made up of many cells called as vessels. Many pipes are joining together. Each cell has a lignified cell wall and large central cavity. Why is central cavity more? Because big pipe, so cavity is much more higher, bigger here. More transport of water is happening. Bigger cavity. So compared to your tracheids, much more bigger cavity. Now the pipe which is going to gutter, does it have a tap? Someone will go and close the tap, open the tap, gutter water is going. No tap. There is no tap at all. So just like how gutter pipe does not have any tap, here also you will never find tapering ends. Can you see tapering ends here? No tapering ends here. No tapering ends at all. Okay. No protoplasm, dead cell. Interconnected through perforations in their common walls. Understand? Here there is some perforations. Perforations are here. Perforations. If there is perforations here, all the water can transport easily into these perforations. Okay. Mori. Gutter means mori. The roadside where all the waste goes, no, gutter. Mori. I don't know what is called in English now. It's called gutter only, I feel. Okay. Now, I told you previously, this was found, the tap in your house. It was found in the case of your gymnosperms and pteridophytes. This one is mainly found in the case of your angiosperms. Now, students, what is tracheids? Tracheids is small pipe. Tracheids is small pipe. Vessels is the big pipe. Tracheids have a tapering end. Vessels no tapering end. Tracheids is unicellular. Vessels are multicellular in nature. Vessels have a larger cavity and it has perforations in the common wall, so water can transport. Vessels nothing like that. Both are dead cells. Drainage. Nice. Drainage. Fancy people. English and all you know full. Okay. Little bit weak in English. So yeah. So students, did you understand the difference between vessels and tracheids? This is a topic which has been a nightmare even for me when I was your age. I'm like, I by-hearted it all my life, but I could never get into it. Only when I made this story back in the day, not now, when I was your age, I made this story back then. And till now, I have never forgotten the difference between a tracheid and a vessel. Always in my head now. Tracheid is small pipe, vessel is a large pipe. Oh God. And I am telling you the same theory which I learnt, and trust me, it will help you. You can remember it. Now here, present in all vascular plants, vessels present only in angiosperms, very exclusive, okay, very exclusive. Narrow lumen, because house, no, we don't need much. Wider lumen, it's going to the drainage directly, okay. Highly thickened, less thickened completely, okay. Do not have end plates, it has end plates are there. Done. Tracheids are vessels. Parenchyma is very simple again. It is just like a normal parenchyma only. Just like a normal parenchyma. Living cell, thin walls, cellulose depositions. Function is what? Storage food. Simple, nothing to study here. Storage of food in the form of your starch or fat and other substances like tannins. Simple. Okay, your parenchyma is very simple. Fibers again. And remember, this is living cell. Okay. Fibers, highly thickened cell wall, obliterated central lumen. What is the meaning of obliterated? Obliterated means completely kalas, fully covered. This is your entire fiber. If this is your entire fiber, the lumen is completely obliterated. This is the meaning of obliterated. Can you see here? This is the meaning of obliterated lumen. Lumen is completely covered by depositions. Obliterated lumen. Okay. Central lumen. It can be septated or separated. What is the meaning of septa formation? It can have a cross wall. Okay, it can have cross walls. Done. Beautiful. Okay. Now students, your xylem is over. Now we'll go to phloem. After that we'll take a break. Don't worry. Time is there still. Night is young today. Full. Okay. So xylem, we learned about primary, secondary. Primary will have protoxylem and metaxylem also. I'll tell you this later on. Secondary xylem is later formed. Okay. And in protoxylem, metaxylem also, protoxylem is first formed, metaxylem is later formed in primary xylem also. Now students, ready for a small concept learning. Many questions will come from here, and if you learn this concept right now, many other concepts will become very easy for you. Very easy will become later on. Ready all of you? Ready? Magic you see now. Okay. We have the primary xylem, very important. PYQ alert. It is always primary xylem and not secondary xylem. Not secondary xylem. Okay, not secondary xylem. Always primary xylem. Primary xylem can be in the form of exarch or endarch or exarch. Endarch or exarch. Understand the difference right now. What is going to be endarch? What is exarch? Okay, what is exarch and endarch? Understand. Okay. In the case of endarch, first of all, it is found only in the case of your stem. Only in the case of stem. And in the case of endarch, protoxylem. Protoxylem is towards the pith. This is the pith, right? Here we have the pith. Protoxylem is towards the pith. Metaxylem is away from the pith. Metaxylem is away from the pith. Now you will tell me, sir, difficult to remember. Difficult to remember. Small trick I have, listen. For example, how do you understand exarch condition? RX. How many of you know the RX bike? How many of you know the RX bike? Okay, RX. RX bike is there. So what is R? R is for your root. X is for your exarch condition. Exarch condition. And the who is X? Who is X, students? X is person who is outside your life now. X is a person who is outside your life. Outside. So meaning of, can I also call outside as a periphery? Periphery. There is a X whose name starts with P, who is outside your life on the periphery. To root exarch, this P stands for protoxylem. Protoxylem is on the outside. Protoxylem is on the outside. Protoxylem is on the outside. So exarch condition in the case of root Rx, in the case of roots, protoxylem is towards periphery. So if you can remember this part, like X is a person who left your life, outside, periphery, protoxylem. Protoxylem is towards the periphery, that is towards the outside here. Protoxylem towards the outside, and metaxylem on the inside, that is called as your exarch condition. That is called as your exarch condition. Okay. Now, what is endarch? In endarch, it is present in the case of your stem, present in the case of stem, and also endarch condition, your protoxylem. See, here it was in this outside. In exarch condition, it was outside, periphery. So, in the case of your endarch, it will be towards center or towards the pith, or towards the pith. Okay. And metaxylem will be periphery. Metaxylem will be periphery. Simple difference. Done. Done. Okay. So exarch, root, protoxylem towards outside. Protoxylem towards outside. Okay. Now we start with the phloem now. Start with the phloem now, students. Now, simple question for all of you. Tell me, tell me students, the main function of a phloem. Go. Tell me students, what is the main function of the phloem? Very difficult question, all of you, difficult question. What is the main function of a phloem? Go. Time starts now, all of you. Time starts now, all of you. Quickly. What is the function of phloem? You will tell me, sir, the main function of phloem is conduction tissue. First of all, it is a conducting tissue. It is a conducting tissue. Helps in conducting food. Helps in conducting food. For example, students, this is the branch. This is a leaf canopy. All the food is produced here. Okay. On the top also we have, for example, on the top we have some, for example, here you have a flower. Flowers are developing. Here. Okay, flowers are developing. Here. Now, if the food is produced in the case of your Food is produced in the leaf. And the food from here, from the leaf, it will also go to the shoot. The food will also go to the entire root system. Entire root system also. So can I tell the transport? Can I tell the transport of the food with the help of phloem? Transport of food with the help of phloem is bidirectional. It is bidirectional. Bidirectional in nature. Bidirectional in nature. Now, under your phloem, we have something called as sieve tubes. We have something called as sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Now, again, the question to all of you: tell me how many of the following are living and how many of the following are dead now? Here, quickly tell me in the chat. How many of the following are living? How many of the following are dead tissues? Quickly in the chat. Quickly. Are living and dead. Simple question, no? Simple question. For example, your sieve tubes is going to be your living. Companion cell is going to be living. Parenchyma obviously living. Only phloem fibers is going to be non-living. What is the ratio of living to non-living now? The ratio is very simple. The ratio of living is going to be 1. Living is going to be 3:1 again. 3 being the living right now. Living. And 1 being the non-living. 1 being the non-living right here. Living to non-living. Okay. What happened? Cricket Club website news. Tell me what you didn't understand. Okay. So living versus non-living. Very simple, right? Very simple. Beautiful. Now students, how many of you have confusion between? How many of you have confusion between the sieve tubes as well as companion cells? Raise your hands quickly, all of you. Raise your hands. How many of you have a difficulty in remembering all of this? I have added entire thing on one slide for you to understand. Very simple. Very simply you can understand this. Okay. First of all, I will show you the diagram and teach you with the help of diagram. Okay. No, I'll show you NCERT diagram only. Wait. All of you can see the NCERT diagram now? Yes, NCERT diagram. Now, if you look here in your phloem, right, in the phloem, there is something called as companion cells here. Can you see? Companion cells. And we have the sieve tube elements here. Sieve tube elements here. Two things are here. Okay. Now, if we can notice here, your sieve tube elements are the middle part here. White color part is the sieve tube, which is elongated cells with perforations. Can you see here? With sieve pores. This is called as sieve pore. Sieve pore right here. And if you notice, your sieve tube elements does not have any nucleus at all. Okay. But at the same time, we have companion cells. Can you see the companion cells have nucleus? Companion cells. This is the companion cell. The pink color one is companion cell. The companion cell has nucleus here. Companion cells have nucleus. They can also interact like this. They can interact. Your sieve tube cells and companion cells can interact sideways also. They interact sideways also. Okay. Now understand. If you look at the diagram, you can tell me, sir, they are long tube-like cells. Here. Long tube-like cells arranged longitudinally. Yes, we have arranged them longitudinally. Okay. Association with companion cells. Yes. Now, why are there association there? We study them together. Right? We study them together. The reason being that they are also called as sister cells. They are called as sister cells. Okay. Now understand, the end walls are perforated in the sieve-like manner. Did we see that here? The end walls are perforated. Can you see? The end walls are perforated here. End walls are perforated here. Can you see? End walls are perforated. You can see here also. End walls are perforated in the case of your sieve tube elements. Sieve tube elements. End walls are perforated here. Perforated. Nice. Okay. Now, mature sieve elements has peripheral cytoplasm and large vacuole and lacks nucleus. So it has large amount of peripheral cytoplasm. Now, what is the meaning of peripheral cytoplasm? Understand. Now, if it is transporting food, if this particular tissue is transporting food, if we have something in the middle, will it restrain the flow? For example, all the food is being transferred here. If there is some form of blockage, you cannot pass through the food easily. So what do? We transport the cytoplasm. All the cytoplasm will be here. All the cytoplasm will be here. It's called as peripheral cytoplasm. It is called as your peripheral cytoplasm. Peripheral cytoplasm. Peripheral cytoplasm in the sites. Okay. Now, if the nucleus was there, it would have been completely blockage then. So the nucleus is completely absent here. Nucleus is absent here. Nucleus is completely absent here. So this is how the structure looks like. Peripheral cytoplasm, nucleus is absent, end walls are perforated. Can you see here? The end walls are perforated here. The end walls are perforated. End walls are perforated, arranged longitudinally, arranged, long tube-like structure, long tube-like structure, done. Now students, sir, you might ask me, sir, if it does not have nucleus, how is every single function controlled here? For that we have a sister here. For example, how many of you have older sister or older brother? If you don't have the money, you'll ask them. They'll give you free money. Right? They'll give you free money. So similarly, we have one more sister here. We have a sister called as companion cell. Sieve tube has a sister called as companion cell. Since sieve tube does not have any nucleus, it cannot control the functions of the pressure gradient or anything. The entire function of your sieve tube element is controlled by companion cell. Clear? So the main function of your transport of food, is happening with the help of sieve tube. Then what is the function of companion cell? The main function of companion cell is maintaining the pressure gradient in the sieve tube. Why? Because the sieve tube does not have a nucleus. Okay. See, specialized parenchyma cell closely associated with the sieve tube. Okay. The sieve tubes and companion cells are connected by pit fields present between common longitudinal walls. There are pits there through which they can connect it. Okay, which are connected. That is all about your companion cell. Xylem, primary xylem, right? Primary xylem is present in both monocots and dicots. Primary xylem is present in both monocots and dicots. Okay. Especially the apical primary xylem is present. Now. Okay. Any difficulty here to understand? We understood here sieve tubes are long tubular cells, peripheral cytoplasm, no nucleus, end walls are perforated. Sieve tube elements. At the same time, your companion cells has nucleus. It can take care of the sieve tubes by maintaining the pressure gradient. It is closely associated with the sieve tubes. See here, students, see here. This is companion cells. The pink color one is the companion cell. Here, right here. Okay. This is basically your, see I told you, long tap-like structure, small tap, tracheids. Then we have vessels, large pipe vessels. Okay. Now the next one is Phloem parenchyma, phloem fiber, very simple, very easy to understand. Phloem parenchyma, all of you know the parenchyma, living cell. Parenchyma is living cell. Main function is what? Storage. Always storage main function. But there's one point which you need to remember here is it is absent somewhere. It is absent somewhere. That is, it is completely absent in the case of monocots. So remember, so students, phloem parenchyma is absent in the case of monocot, and this right here is a PYQ. Previous year question, they'll keep asking you. Absent in the case of monocots. Since it is a parenchyma cell, elongated, tapering cylindrical cell, dense cytoplasm nucleus because it's a parenchyma, right? Plasmodesmal connections exist between the cells. It means the cytoplasm is connected here. Okay. What is the function? Storage of food again. Storage of food is the main function. Always. Fiber is always dead, right? Made up of sclerenchymatous tissue, dead tissue. Yes. It is absent in primary phloem, present in secondary phloem. So can I tell it is a type of your secondary tissue also? Yes, secondary tissue. Okay. Megan, I told you, right? Absent in primary phloem, present in secondary phloem. Secondary phloem, later on it is formed. Elongated and unbranched, tapering ends. Pointed needle-like apices, at maturity lose protoplasm and becomes a dead cell basically, dead cell. Students, I would highly advise you to download these notes, make the following points and then learn it, because it can get confusing very fast. Okay, so learn point by point, everything. So we learned about your phloem, primary xylem, primary phloem, secondary phloem. Okay, here we have protophlloem, metaphlloem similarly. Question is right here. I told you, gymnosperms lack xylem, vessels, because vessels are only found where? Vessels are only found in the case of your angiosperms, only in the case of angiosperms, right? And there are something called as albuminous cells. I'll show you the NCERT page. I'll show you the NCERT for that. What is the answer, students? What is the answer? So we are done till now here. Can you see here? We learned about your collenchyma, parenchyma. We learned here spherical, oval, round, polygonal, elongated in shape, closely packed or have small intercellular spaces. We learned here. We learned about your collenchyma also. It occurs in layers below the epidermis, in hypodermis, right? Hypodermis. We learned about sclerenchyma also, fibers and sclerets, and where are they found? We learned about your xylem, tracheids and vessels, elongated. Can you see here? I think all of you are able to see this, right? Gymnosperms lack vessels in their xylem. So gymnosperm will lack vessels, completely lack the vessels. Okay, vessels are absent. Tracheids are elongated tube-like cells with thick lignified walls and tapering ends. Okay. Where do you find vessels? Vessels are mostly found in the case of your angiosperms. Endocrine starch condition. In phloem also you see your phloem in angiosperm is composed of sieve tubes, companion cells, phloem parenchyma and phloem fibers. So all the 4 different types of phloem is present in the case of angiosperms. But in the case of gymnosperms it is not found. Let me show you here. In the case of gymnosperms have albuminous cells and sieve cells. They lack companion cells. So in the case of gymnosperms, they do not have a companion cells at all. Very, very important line of NCERT. Okay, this is what I wanted to show you. Okay, so gymnosperms have albuminous cells and sieve cells. They lack the they lack sieve tubes and companion cells. Sieve tubes and companion cells are absent in the case of your gymnosperms. At the same time, even vessels are absent in the case of your gymnosperms. Right here. What is it? Gymnosperms lacks vessels. 3 points, please remember that. Please remember that. Students, for this we have to follow both old and new NCERT both. Old NCERT for whatever we learned till now in the last 1 hour for 2 hours 45 minutes. Whatever we learned, that is for your 2 hours. What we learned old NCERT you have to learn. They'll ask you examination. See here, this is a NEET 2024 question. NEET 2024 question. Till now there has been no change in syllabus. No change in syllabus. So you have to follow the old syllabus itself of NEET 2024. Yes. So first statement is wrong obviously because it says 𝐂𝐡𝐞𝐤 is dead. We know it is living. And we know second statement is correct. Gymnosperms lack xylem vessels. Vessels is absent. Sieve cells is absent as well as companion cell is absent in gymnosperms. The given figure with reference with the anatomy of plants represents. What is the structure? Big pipe. Tell me the answer. Big pipe. See, trachea was smaller pipe. Trachea was smaller pipe. This is bigger pipe. No. What is this, students? Bigger pipe. What do you call the bigger pipe here? Bigger pipe. Exactly. The vessels. Vessels. Next question, students. Next question. It's again NEET 2024 question. Companion cells help in maintaining the pressure gradient in the sieve tubes. Correct. You are supposed to tell which of the following are correct. Gymnosperms lack vessels in their xylem. We learned just now. Gymnosperms lack the vessels. Vessels are absent here. Vessels are absent. Absent. In case gymnosperms. Xylem vessels are devoid of cytoplasm. Vessels dead, right? Vessels no cytoplasm. Correct. Xylem vessels are devoid, dead. Yes. Xylem fibers may be septate or aseptate. We just now learned fibers. We learned, right? We learned about fiber. Xylem fiber may be septate or aseptate. You can have septa formation. Yes. A mature sieve element in phloem possesses cytoplasm, vacuoles and nucleus. Yes, everything is there. It has nucleus also. We learned here. We learned here. Where is the NCERT for that? See here. A mature sieve element possesses peripheral cytoplasm, large vacuole but lacks nucleus. So peripheral cytoplasm, large vacuole and lacks nucleus. So what is the answer now? Tell me, students. What is the answer? It's also correct. ABCD, right? ABCD. Done. Look at this question from NEET 2024 again. Do you realize how many questions have come from this part itself? 3 to 4 questions from this part. Okay. In 𝐂𝐡𝐞𝐤, cell walls are thickened at corners due to depositions of cellulose, hemicellulose and pectin. And pectin is there. Cellulose, hemicellulose and pectin. Okay. 𝐂𝐡𝐞𝐤 constitute of lignified cell wall and possesses pits. See, I told you, lignin depositions is 𝐂𝐡𝐞𝐤. Pectin depositions is going to be mostly for your 𝐂𝐡𝐞𝐤. Only cellulose in the case of your parenchyma. So, tell me the answer. Both are true. Yes, both are true. So, it will have all three actually. Cellulose, hemicellulose and pectin. Okay. Next question. Companion cells are closely associated with. Who is the sister of? Who is the sister of 𝐂𝐡𝐞𝐤? Two students. Companion cells are closely associated with. Who is the sister? Very easy question, students. Very easy questions. It's going to be your sieve elements. It's going to be your sieve elements. The chief water conducting element of xylem in gymnosperms is gymnosperms. See, in the case of gymnosperms, do we find vessels? See, fibers gone. Do you find vessels in the case of gymnosperms? we only find tracheids. So answer is 4. Answer is 4. Vessels is absent in the case of your large pipe, is absent in the case of gymnosperms. Oof. And with that, the entire tissue part, it could be your friends. This is the entire part we did today till now. Okay, entire thing is over. Can you see here? We start off with the entire tissue, meristematic tissue. We did diagram, axillary bud, intercalary, primary meristem, secondary lateral meristem, simple tissue, parenchyma, collenchyma, sclerenchyma. Then we did about your xylem, tracheids, vessels, xylem fiber, xylem parenchyma. Yes. Your meta, endark and exarch condition, endark in the case of root, endark in the case of stem, exarch in the case of root. Then we did the companion cells for the phloem, sieve tubes and companion cells, phloem, parenchyma and phloem fibers. Everything is done. Now we have to start with tissue system. Tissue system, where we study about epidermis, cortex and stele. Epidermis, cortex and stele. Cortex, that is ground tissue, and the entire stele part. And then the main part of this chapter where many questions will come from, which you cannot by heart any of this. If you sit and by heart the entire of this, you will go mental. Okay, so this thing is going to be very difficult to by heart later on. So you don't have to by heart. I will write you notes. I'll make you write down notes for every single section. Every single section I will make you that write down some notes. Okay, for this entire part, which we will be doing today only. Okay, now we will take a 15 minutes break and we'll come back and then we will start all this. Okay, then we will start. Today we will finish the entire chapter only, students. Then leaf is there, that's all. Root, stem and leaf is there. Okay, now we will take a, what is the time now? 8:45. We'll start at 9:05. Okay, class resume at 9:05. Class will resume at 9:05. Okay, so all of you quickly go eat and come something. This is dinner break. Dinner break. Okay, now after the break is over, we will start a tissue system. Very simple again, very simple it is. We'll talk about epidermis, cortex and stele. Then all the different sectioning, students. Sectioning, trust me, learn the sectioning from me, right? You will never forget. Okay, you'll never forget. Okay, now we will come back again. Bye, take care all of you. Now, students, welcome back to all of you. Welcome back. And now we'll be starting off with the second part of the chapter, that is going to be the entire tissue system. Okay, I don't know where I kept the pen now. Great. 1 second. Yes, we are back. Yes, we are back all together. Okay. Now, students, you need to understand here is till now what we have done. First of all, let me tell you. Till now we have done something called as meristematic tissue. Completely done. Primary and secondary meristem based on origin. Based on location, we have intercalary meristem, apical meristem, and lateral meristem. Then we start off with your simple tissues. Simple tissues where we studied about your parenchyma, collenchyma, sclerenchyma. Complex tissues, xylem and phloem. Every single part is done and dusted now. Now we will start something called as the entire tissue system. Okay. In tissue system, multiple tissues are coming together and making a system here. They're working together, coordinate together, functioning together. Okay. Now, under your tissue system, we have 3 different categories. The first one is called as your epidermal tissue system. Second is called as your ground tissue. And the third one is called as the vascular tissue. Vascular tissues or vascular bundles. Now we will be studying each one of them. That is epidermal, ground tissue, and vascular tissue system. Not that tissue, different tissue this is. Okay, different tissue. Now, if you can see this diagram, all of you, this is your epidermal part. On this epidermal tissue system, do we have the epidermal appendage? Yes, on the top we have the epidermal appendage. The meaning of appendage is an extension. Appendage means a thin extension. From the epidermis we have appendage coming out. Then we have the entire epidermal cells here. Here, can you see this one? This one right here is the epidermal cell. Okay. Complete single layer of epidermal cells. Then we also have in the epidermal cells, we have something called as stomata. Okay. In epidermal system, students. Now, each one of them we will study separately. First, we will talk about epidermal appendages. Okay. Now, there are 2 types of epidermal appendage. All of you know this. The first one is called as root hair. Now, students, I want everyone to wind back the time and think about morphology chapter. In the morphology chapter, did we study the structure of root? All of you tell me. In the morphology chapter, did we study about the structure of a root? Yes. This is the main taproot. And in this taproot, this is called as the region of meristematic growth, region of elongation, and this was called as your region of maturation. Now, in the region of maturation, there was a beard coming out, no? Yes. In the region of maturation, we had something called as root hair development. Now, tell me, students, this root hair right here, this is the root hair. Is it developing from the outside or inside? Tell me, students, is it a exogenous origin or endogenous in origin? Tell me in the chat quickly. This root hair right here, can I call it as a exogenous in origin? It is exogenous in origin and it is unicellular in nature. And what is the function here? The main function is increasing the surface area. That is the root hair. Okay. Then what is the trichome? Students? What is the trichome? Now, students, all of you, have you, any one of you tried to touch a tomato plant? If you were to touch a tomato plant, on the stem, entire stem, on even on the sepals, right? Even on the flower also, the sepals, we can find small hair-like structure. Even in the case of pumpkin also, you might have noticed, pumpkin plant is growing. On the leaf, you will see a small hair-like structure on the leaf. You will see a small hair-like structure even on the stem. Yes, something like this. On the stem, we can see small hair-like structure. That hair-like structure is called as your trichome. Trichome is basically the stem hair. Stem hair. And apart from that, your trichome is actually, it can be multicellular, or it can be unicellular also. Single cell or multiple cells. Okay. Now students, all of you tell me, here we told the main function of your root hair was increase in the surface area. Now what could be the function of trichome? Can anyone tell me the function of a trichome? Now students, understand, on the leaf we have the trichome. This trichomes can actually decrease the water loss by reducing the transpiration. They can decrease the water loss. At the same time, some of the trichomes can be very pointed. Like they can be pointed, they can be tough and soft at the same time. Some of them very soft. If you touch it nicely, you can press it also. Nothing will happen to you. Okay. It can be soft also, soft or hard. Okay. Now all of you remember the meaning of septa, right? Imagine this is a trichome. This is a trichome right here. This trichome right here can have septa formation, cross wall formation. So it can be septate or aseptate. And at the same time also, students, understand this, a trichome can also produce something. Like, have you noticed this? There are certain plants, if you touch the stem, your entire hand will become red. Because of the allergic reaction. Because on the trichomes, there are certain secretory nature. It has secretory nature also. So trichome can produce certain substances which can harm. So basically it is a defense mechanism. Yes or no? It is a defense mechanism right here. Defense mechanism. Beautiful. So that is the difference between our root hair and trichome. Any doubt, quickly shoot in the chat, all of you. Shoot. Root hair is exogenous. This was exogenous. Unicellular, multicellular, region of maturation, increase in the surface area, more absorption. At the same time, trichome is stem-like, stem hair, multicellular, unicellular, reduce the water loss, tough and soft, septate or aseptate or secretory in nature. Done. Now students, now is the small game all of you. Now all of you understand. Look at this diagram of right here, this layer right here, and tell me, what do you notice? I'll ask you simple questions now. What do you notice? Is it the outermost layer or is it present on the inside? Tell me, all of you. See, this is how I teach botany. Okay, you look at things, you understand. You don't have to buy any of it. Tell me, is it outermost layer? You're like, yes sir, it is the outermost layer. Since it is the outermost layer, can I tell it main function is going to be protection? This is for your epidermal cells. Okay, epidermal cells is protection. Okay, it is protection. Main function is protection. Now, do you see this is the first layer? Is it a single layer or multiple layers? Look at the diagram, all of you. Do you see a single layer or is it made up of multiple layers? Tanuja, tell me answer. Tanuja, tell me. Is it single layer or multiple layers? All of you tell me. It is going to be single layer, like all of you. Okay. It is single layer. Okay. It is single layer. Now, do you see any type of intercellular space here? Anyone? Do you see any space here in the first layer? Do you see any space here? Any form of intercellular space here? You're like, no sir, I do not see anything. Intercellular space is absent. Intercellular space is completely absent. Okay. Intercellular space is completely absent. Now students, It is made up of parenchyma. First of all, it is made up of parenchyma. Even though it is made up of parenchyma, see students, all of you tell me, does every single parenchyma cell has to be photosynthetic? Yes or no? So this is the problem with NCERT diagram also. Every single time you think of parenchyma, you think about the green color cells. That is not the case always, because the main function of parenchyma is storage or packaging. Green color one is chlorenchyma. Green color one is chlorenchyma. So don't get confused. Every single parenchyma is not photosynthetic in nature. It is not photosynthetic in nature. Do not get confused. Only chlorenchyma is photosynthetic in nature. Generally speaking, a particular parenchyma main function is packaging. Chlorenchyma, aerenchyma, palisade, spongy parenchyma, all that is separate categories. Okay. It can be, the color which they are showing you is for your own, just to understand, that's all. Okay. So students, generally here, 6th point, chloroplast is absent. If chloroplast is absent, can it do photosynthesis? All of you tell me in the chat now. Think simple logic. All the students who are watching it recorded also. Think logically. If it does not have chloroplast or chlorophyll, specifically speaking, can it do photosynthesis? Obviously not. It cannot, right? But except chloroplast is absent except at a site called as, can anyone tell me, there is a small site where it does not have any, it has chloroplast. It looks something like this. Here. What are these? Except at a site called as guard cells. Except at the guard cells, there will be no chloroplast at all. Except the guard cells. Clear? All of you understood? Except at the guard cells, every single place there will be no chloroplast whatsoever. Now students, tell me, this is a structure present on the outside, on the leaf surface, right? Leaf surface. Now, how many of you notice this? How many of you know colocasia plant? Colocasia. How many of you know? Large leaf will be there. Large leaf. On that, it looks something like this. Colocasia plant leaf. Large leaf will be there. On this large leaf, if you add water, the water will nicely slide off. You can take the colocasia and make bhaji out of it. Cut the plant and make bhaji and eat also. Little pointy it will be, but yeah. What do you think that is? Why do you think if you add a drop of water on the colocasia leaf, or generally any of the leaf, most of the xerophytic leaves, if you add water, the water will slide off. It's like, don't touch me. Water is like, don't touch me. It will slide off on a slide. Why do you think that is happening? What is so special about the leaf that water cannot hold there? Water cannot touch the leaf. Why? Because it has a waxy layer. Can anyone tell me what is the waxy layer I'm talking about here? Okay. It has a waxy coating, 7th point, right? It has a waxy coating on the epidermis. Waxy coating on the epidermis called as cuticle. Called as your cuticle. Okay, cuticle. Now students, tell me, this cuticle is present in the case of your epidermis. But is it present in the case of root? Will the cuticle be present in the case of root? Obviously not. Okay, so this cuticle will be absent in roots. Okay, last point students. Till now I think everything is easy, right? All the 7 points are very easy till now. Last point is that they have large vacuoles. 8th point is they have large vacuoles. Large vacuoles. Large vacuoles. Done. That's all. It's epidermis. Nothing to break your head. Colocasia. C. Colocasia from C. Colocasia. Okay. So outermost layer, protection, single layer, interstellar space is absent, made up of parenchyma. Chlorenchyma is absent except at the guard cells. Large vacuoles, waxy coating, cuticle, absent in roots. Absent in roots. That's all is epidermis. Nothing to learn more than that. Okay. Now stomata. Diagram I'll show you. Stomata, very important diagram I'll show you. Okay. In stomata, all of you, listen very carefully. This is the structure of stomata. We have two guard cells. We have the two large guard cells here. This is a guard cell. This is a guard cell. Okay, this is a guard cell. Can you see under the microscope also? They are the two guard cell. This is called as stomatal pore or stomatal opening. Stomatal pore or opening. Around the guard cells, there is epidermal cells. Around the guard cells, there are certain epidermal cells which become very special later on. I'll show you. There are special epidermal cells. The special epidermal cells around the guard cells, these cells, the special epidermal cells around the guard cells are called as your subsidiary cells. They are called as what? Subsidiary cells. Subsidiary cells around the guard cells. Okay. Now, in the guard cell, students, the inner part is much more thicker here. It is much more thicker on the inner side. The inner part is much more thicker. Why? Because it has to hold the pressure gradient, right? For example, the stomatal pore is opening. The stomatal pore is opening. If stomatal pore is opening, it has to, water is going out. So this part, the inner part is much more thicker always. It is much more thicker compared to the outer part. Now I'll tell you one more small thing here. That is subsidiary cell plus the stomatal pore plus the guard cell. Entire thing is called as stomatal apparatus. All three together, subsidiary cell, stomatal pore, guard cell, all three together is called as stomatal apparatus. Stomatal apparatus. Now students, in examination many times I've asked you is the structure of stomata. Structure of the entire stomata structure, entire structure of stomata. In the case of monocot, it is dumbbell shaped. Can you see? Dumbbell shaped here, dumbbell shaped, and here we have bean shaped, which we drew right now. The bean shaped in the case of dicot. Bean shape is equal to in the case of dicot. Done. Epidermal, done students. So what we learned in epidermal, we learned about the stomata where we have chloroplast and the shape of it we learned here, and we also learned here something called as stomatal pore, guard cells, subsidiary cells. All three together is called as stomatal apparatus, and every single time water is entering, the stomatal pore, the guard cell will become turgid and stomatal pore will open. As soon as the water is gone, they will become flaccid and the stomatal pore will close. So imagine it's like something like this. These are the two guard cells. Every single time water is entering the guard cells, it becomes turgid. And the water is gone. Once the water is gone, the guard cell will completely close off. Okay, that is flaccid condition. We learned about your epidermal tissue, epidermal cells entirely. We also learned about epidermal appendages, root hair and trichome. That was basically the outer part. Now we will study this part. You see middle part. Middle part is called as your middle ground or the ground tissue. Also called as your ground tissue. Now what is the definition of ground tissue, students? Anything between, this is epidermis, here we have steel. This is epidermis and steel. Whatever is present in the middle, right? Whatever is present between your epidermis and steel or the vascular bundles, or the vascular bundles, the middle part is called as your ground tissue system. Middle part is called as what? Ground tissue system. All tissues except epidermis and vascular bundles. All the tissues except the epidermis as well as the vascular bundles, anything in the middle is called as your ground tissue system. Ground tissue system. Okay, students, ground tissue system. Your entire ground tissue system is mainly made up of few things, main few things. First one is called as your middle cortex. We have the cortex here. Then we have the pericycle. We have the pericycle. We have the pericycle. Then we have in the middle, then we have what we have here. In the middle we have the pith. And we have the pith here. Okay. Now students, understand your cortex is made up of three main things. Cortex has multiple layers. Below the epidermis, what do we have, students? Below the epidermis, where we find 𝐂𝐡𝐞𝐤. What is that? Something called as hypo, right? Below the epidermis, we had something, right? What do you call that? That is called as your hypodermis. Hypodermis is part of your cortex. The innermost layer, the innermost layer of cortex is called as, it is called as what? It is called as endodermis. Endodermis. The innermost layer of cortex is called as your endodermis. Cortex, then we have above layer, that is hypodermis, endodermis. In the middle we have cortical layer. I'll tell you later on. Cortical layer later on. Okay. Whereas after endodermis, what do we have? After endodermis, we have the pericycle after this. Pericycle after this. Okay. Now students, we have one more small thing to learn before we go to all the sectionings. Can we do that now? We have one more small thing before we go to the sectionings. The small thing which I am talking about here is the vascular tissue system. That is the arrangement of xylem and phloem. One small thing is there. Understand the small part. Everything will become easy for you later on. Okay. For example, can we start, students? Can we start? The small thing about vascular system is for example, students, for example, here we have xylem, we have phloem, xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem, xylem and phloem. From the center, can I tell each vascular bundle, xylem or phloem, phloem or xylem, phloem, xylem, each of the following vascular bundle is its own radii, yes or no? Every single, you know radii, right? Maths, radius, diameter, radii and diameter, right? So each and every vascular bundle is in an alternating manner. It is in its own radii. Such an arrangement of vascular bundle is called as your radial arrangement. It is called as what? Radial arrangement. Radial arrangement, and it is seen in the case of roots. Okay, done. Now, what about this one? Can you see? Xylem, phloem, xylem and phloem, xylem and phloem, xylem and phloem. Now, can you see here in the second type of arrangement, xylem and phloem are present in the same radii now. They are not presenting alternatively. Both xylem and phloem are present in the same radii right now. So, this type of arrangement This type of arrangement is called as your conjoint. They are joined together. They are joined together here. So conjoint. And conjoint is mainly seen in the case of your leaf and stem. In the case of leaf and stem. Now students, understand, this conjoint is actually of two types. Conjoint is of two types. Okay, understand. This conjoint also is of two types now. Okay, understand. Now in the first type, can you see xylem, phloem, xylem, phloem, xylem and phloem, and xylem and phloem. Now can you see here, xylem is present towards the pith. Can you see here, students? Xylem is present towards the pith. Xylem is present towards the pith. Here conjoint means both can interchange. In conjoint, both can interchange. It's called conjoint because they're present together. But in the case of your collateral, your xylem is towards the pith. Xylem towards the pith. Here. Xylem is towards the pith here. Can you see here? Xylem is present towards the pith. Here. Now, in the second one, it's called as bicollateral. In the case of your bicollateral, what happens? In the case of bicollateral, we have, let me change the color for this. Phloem, xylem, phloem. Phloem, xylem, phloem. Phloem, xylem, phloem. Phloem, xylem and phloem. In this other case, which is called as bicollateral, your xylem is sandwiched. Xylem is completely sandwiched between the two phloems here. Okay, between the two phloems here. It is completely sandwiched here. Okay, this is collateral, conjoint, radial. Now again, your each vascular bundle is of two types now. It is of two types. Understand? This xylem and phloem arrangement is there, right? We will magnify it. Let's magnify this one now. Let's magnify this one. Now between xylem and phloem, if between xylem and phloem there is a presence of a cambium, then it is called as open type. Why? Because cambium is present. And if cambium is present, it can undergo secondary growth. Secondary growth is possible. If cambium is present, type it on top and box will write off. If cambium is present, then secondary growth will happen. Okay. Now in this below one, there is no cambium. Here. No cambium. And also no secondary growth. Here. Okay. And this condition is called as closed condition, completely band, open and closed. And this closed condition is mainly found in the case of your monocots. This is found in the case of dicots. In the case of your dicots, okay, dicots. And also this is present in the case of your leaf and it is present in the case of your stem and root. In monocots and dicot leaf, and also dicot leaf. Now why dicot leaf? Because a leaf cannot undergo secondary growth. A leaf will not show secondary growth at all. A leaf is always closed. A leaf is always closed. An entire monocot is closed. In dicot, stem and root is open and 𝐤𝐤 is present, and that is why a stem will show secondary growth, a root will also show secondary growth. Okay, I hope students, do you want these notes? Do you want these notes? Very nice, no? Entire vascular tissue system in one slide. Entire vascular tissue system in one slide right here. Radial, every single xylem bundle and phloem bundle its own radii, roots. Conjoint, leaf and stem, they present together, conjoined, present together. Okay. Bicollateral, it means phloem, xylem, phloem, phloem, xylem, phloem. Collateral, xylem towards the pith, phloem away from the pith. Open, it means 𝐤𝐤 is present. Close, 𝐤𝐤 is absent. No secondary growth. Secondary growth will happen here. In the case of dicots, open is there, stem and root. In the case of monocot, we find the closed and also the dicot leaf, because dicot leaf does not show secondary growth at all. Done students? Beautiful. Students, I would advise you, I'll tell you one thing. I'll give you the notes, but promise me you will make your own notes from these notes. Don't study directly from here. It's my request to all of you, because let me tell you, you can have all the notes in the world. Krishna, I'll share you. But unless you sit and make your own notes, learn, do something called a self-study, nothing will happen. You can spend 10 hours learning in a coaching center. You can spend 5 hours on YouTube every single day thinking and learning, I'm learning. But unless you sit at the end of the day, unless you do something called a self-study, you will regret it later on. I've spoken to many students. They told me, sir, I study 10 hours a day. I study 15 hours a day in coaching center, in YouTube. How many hours do you self-study? 20 minutes, 30 minutes. And some students actually told me they only do self-study in the weekend. That sound, students, that will never work out. Let me tell you honestly, whatever you learn, right, whatever you learn from me, unless you sit yourself and revise the entire thing what I've told you today, trust me, if you do not do that, you will forget everything by the end of this weekend. Today is Saturday, Monday, Tuesday, gone. Out of your mind, gone. Why? Because it is science. You have something called a short memory now. If you want to make the short-term memory into solid memory, you need to revise. You need to write down the notes yourself and make your own notes from there. So unless you sit and revise on your own, all this is meaningless. I'm telling you now only. I've told this in all the classes. Sir, initiation of lateral roots and vascular 𝐤𝐤 during secondary growth takes place in the cells of your pericycle. Coming up. Meghana, wait. We will come. It is there. Pericycle is coming. Have some patience. Okay. Now, with that being said, that was the end of tissue system. Okay, why are these notes coming again? I have to delete some slides here. See what is given your NCERT. Epidermal tissue system. Epidermal tissue system forms the outermost covering. I told you, outermost covering of the whole plant body, comprising of epidermal cells, stomata and epidermal appendages, trichomes and hair. The epidermis is the outermost layer. All of you learned that. Outermost layer in the primary plant body. It is made up of elongated, compactly arranged with cells, parenchyma cells. Parenchyma cells. Continuous layer. A single layer, continuous layer. Epidermis is usually single layer. Epidermal cells are parenchymas, I told you already. Small amount of cytoplasm lining in the cell wall and large new vacuole. We learned that every single point we have mentioned here. The outside of the epidermis is often covered by a waxy layer called as cuticle. Okay, prevents the water loss. Cuticle is absent in the case of roots. Done. Stomata are structures present in the epidermis of the leaf. Done. Stomata regulate the process of your transpiration and gas exchange. They have something called as guard cells. Stomatal pore, dumbbell shaped in the case of your grasses or monocots. Grass or monocots, we have the dumbbell shaped. Bean shaped in the case of your dicots. Okay. Subsidiary cells. What are subsidiary cells? They are basically what? Epidermal cell. Subsidiary cells are epidermal cells in the vicinity of the guard cell. Around the guard cell, we have the epidermal cells, which become specialized to make the subsidiary cells. Stomatal aperture, guard cell and surrounding subsidiary cells, all three together make up the stomatal apparatus. This is what is given you in NCERT also. Trichome, see, root hair is unicellular, I told you, unicellular, help in absorption surface area. Trichome are usually multicellular, branched or unbranched, that point is missing. Soft or stiff, I told you, soft or hard. Yes. They may even be secretory, I told you, produce something, allergic reaction. Yes. Trichome help in preventing water loss due to transpiration. Every single point done. Okay. We have the they mostly have the parenchyma cells, cortex, pericycle, pith and medullary race. All of them are part of your ground tissue. Pith, pericycle, I didn't mention this, I guess. Cortex, pericycle, pith and medullary race. Okay. And then we have mesophyll cells. Open we learned, closed we learned, radial, conjoint, 𝐤𝐴 is present, it is open. Okay. See a diagram. See the diagram here, all of you. See the diagram. Here we have the radial arrangement. Everyone is on radii. Conjoint. But tell me, is it open or closed? It is closed. This is open. Why? Because 𝐤𝐴 is present. 𝐤𝐴 is present. And this is your conjoint. Also conjoint. Xylem and phloem are present together. Time starts now. Quickly go. It's mostly monocots. All the monocots. Open means what? 𝐤𝐴 is present. Open means between xylem and phloem, 𝐤𝐴 is present. Which will do, secondary growth. In the given figure, which component has thin outer wall and highly thickened inner wall? I told you, thin outer wall, inside is thick. That is basically your guard cells. That's your guard cells, right? And where are the guard cells here? C. Right? C is your guard cells. So option number 4, C is your guard cells. So do you realize? Very simple question. You can understand this. Guard cells. Stomata in grasses, leaves are. Grasses is basically monocot. So what should be your answer? Dumbbell shaped. Cortex is the region found between. Cortex is the middle part between your epidermis and stele. Specialized epidermal cells surrounding the guard cells are called as. We have specialized epidermal cells which become guard cell, around the guard cells we call them subsidiary cells. Vascular bundles in monocot are considered closed. We have called them closed. Why? In monocots, the vascular cambium, the vascular bundles are called as closed. Why? We call them closed because cambium is absent. Because cambium is completely absent. Okay. closed vascular bundle lacks. What does it lack? It lacks the cambium. Such an easy question again. It lacks cambium basically. It lacks the cambium. Okay. In land plus, the guard cells differ from the other epidermal cells in having. What is the difference? I told you. There's a difference between guard cells and other cells in the subsidiary cells. In the epidermal tissue system, guard cells have something which other cells do not have. Chloroplast. Chloroplast. Friends, do you realize this chapter is actually very easy? This chapter, they always ask direct questions. Always they ask direct questions. And if you have learned the lines of NCERT, if you have learned the concept from me, you cannot miss out on a single question. That's my guarantee. Okay, now students, my favorite part of the chapter now. This is going to be my favorite part of the chapter now. Okay, my favorite part. This is my favorite part. That is the anatomy of dicot and monocot plants. Can we start? This is my favorite part. Trust me. The entire chapter, 5 questions, 4 to 5 questions. NEET 2024, you got around 5 questions from here. 5 questions. 5 questions. Not students. Let me tell you one thing. See, weightage is all well and good, but at the end of the day, don't think any chapter is less. Okay. Now, my favorite part is coming up now. Tell me, are you ready for this next part, which is going to be a crazy banger? Okay. So, can we start now? Can we start with the sectioning now? Sectioning of monocot and dicot plants. Okay. Tell me, how many of you don't like this part? Tell me honestly. Raise your hands. Raise your hands. How many of you find this part as, sir, too much to mug up? Too much to mug up. I get confused always. Right? I always get confused between monocot root, dicot root, monocot stem, dicot stem, monocot leaf, dicot leaf. This is very confusing for me. I cannot by heart entire of it. How many of you feel that? Raise your hands quickly. All of you raise your hands. And all of you quickly smash the like button. Mark your attendance. Right? Mark your attendance. And we're going to have a blast now. Next, probably what? 45 minutes. Just 45 minutes. I will make it the easiest part of your life. Challenge. Challenge. All students who are watching it live, all students who are watching it recorded. Challenge is that I will make it the easiest part, and that is my duty here. Okay. So let's make it the easiest part now. Can we start? Now, my way of teaching is very simple. As a botanist, anatomy should be learnt with the help of a leaf sectioning under microscope. Since we do not have all that, we have a substitute here. The substitute here is going to be your entire diagram. Entire diagram is going to be the substitute here. So what you need to do is, whatever you see in the diagram, report to me, I will write here. Simple, simple. This is how I always teach. This is how I always teach. You tell me what you see here and I write here and you will learn it. Simple. Now students, question number 1. Question number 1. Which is the outermost layer? Quickly go. This is how I teach. Always been teaching many years. Tell me what do you see is the outermost layer here. Outermost layer. You will tell me, sir, the outermost layer is the epidermis right here. The outermost layer is the epidermis. Epidermis. Now students, tell me, is the epidermis a single layer? Can you see a single layer or multiple layer? Tell me, is the epidermis, which is technically the outermost layer, it is single layer, right? It is single layer. Single layer. Okay, sir, it is single layer here. Now students, extra point all of you. In the case of root, the epidermis has a separate name. The separate name is called as epiblemma. It is also called as what? Epiblemma. Remember this point, very crucial. Epiblemma. It's called as what? Epiblemma. Now students, tell me, do you find any type of appendage here? Do you find any type of epidermal appendage? Anyone can see here? I'm not able to see. Tell me. Do you see any type of appendage here? Yes, sir. We can see here root hair. We can see the root hair here. We can see root hair. Okay. We can see root hair. Since it is the epidermis, outermost layer, protection, you can write all that if you want. Protection also. Protection. Can I tell it is made up of parenchyma? We learned it in the previously. Parenchyma. We learned it is made up of parenchyma. Now students, epidermis, do you see anywhere any spaces? Do you see anywhere intercellular space anywhere? Yes or no? Tell me quickly, everyone in the chat. Do you see anywhere any form of intercellular spaces here? You will tell me, sir, intercellular space is completely absent here. Intercellular space is completely absent here whatsoever. Now this is the root section. This is the root section. On the epidermis, will you find cuticle? Simple question, no? Will you find cuticle on the root? Cuticle is even required there. Root is underground. Sunlight is not coming. So cuticle is absent. Cuticle is absent. Now you tell me, all of you, it's a root section. It's a root section. Will you find stomata here? Very simple question, no? Will you find stomata? Absolutely not. Stomata absent. Now you tell me, students, out of all the following points, any point you didn't understand, or should I repeat again? Epidermis is called as what? Epiblemma. It is also called as what? Epiblemma. It is called as what? Epiblemma. Okay, epiblemma. Okay. Outermost layer, check. Outermost layer, check. Single layer, check. Root hair present. Protection, parenchyma, absent. Cuticle is absent. Stomata is also absent. Every single point, checkpoint done. If you are able to understand this, quickly fire emoji in the chat, because epidermis is done and dusted. Epidermis is done and dusted. Now students, tell me, which is the second layer do you see in the diagram? Second layer do you see in the diagram? I can see something called as cortex, no? Can you also see? Can you also see the cortex? Yes sir, I can see the cortex here. Cortex. Second layer is called as your cortex. Cortex. Now students, if you look at the diagram, do you see the cortex is single layer or many layers? Tell me, all of you, do you see cortex as a single layer or many layer? Tell me. You will tell me, sir, we can see many or multi-layer. Multi-layer cortex is there. Multi it became multi. Multi-layer cortex. Multi-layer cortex. Now students, this is also made up of parenchyma. Also made up of parenchyma. Also made up of parenchyma. Now tell me, students, do you find intercellular space anywhere here? Are you able to see small white gaps anywhere? White gaps here, here, everywhere. Yes. So intercellular space is present. Intercellular space is present here. It is present here. Okay. Intercellular space is present here. Beautiful. Cortex is done. That's all. Cortex is done. Now tell me, students, in diagram only again, which layer do you see next? After cortex, what do you see? After cortex, I can see one pink color layer here. That is endodermis. I can see something called as endodermis here. First of all, tell me, is endodermis part of cortex? You're like, yes, sir. It is the innermost. It is the innermost layer of cortex. Yes or no? Innermost layer of cortex. Now tell me, students, the pink color layer, is it single layer or multi-layer? Is it single layer or multi-layer? Single layer. I can also do not see any intercellular space here. Intercellular space is completely absent here. Completely absent. Okay. Intercellular space completely absent. What do you notice? What do you notice here? Okay. It is also made up of parenchyma. Also made up of parenchyma. One extra point I'll tell you now. Two extra points. It acts as a checkpoint. It acts as a checkpoint. That is, Anything which is entering the cell. Anything which is entering the cell, it is like, wait a minute, show your passport and enter inside. No passport, no entry. Okay, customs it is. It acts as a checkpoint. It acts as a checkpoint. Varun, don't worry about NEET 2025, difficult, easy and all. If you study all this, if you prepare from now only, everything is going to be easy. Let me tell you on that. Okay, everything is going to be easy. Now students, tell me, here in the endodermis, we have some form of depositions. In the endodermis, the cells are barrel shaped cells. Now, what is the meaning of barrel shaped cells? Can you see this? This is called as barrel shaped cells. It is called as your barrel shaped cells. Cells are barrel shaped here. Cells are barrel shaped here. Okay, barrel shaped cells. Now, in this barrel shaped cells, there is some form of deposition, students. There is some form of depositions. In this barrel shaped cells, there is some form of depositions called as subarine depositions. Subarine sounds very fancy, no? Subarine depositions. And the subarine depositions will end up making something called as Casparian strips. Casparian strips. And these Casparian strips are completely impermeable to water. They are like water, no entry. Whatever you do, I will not let water enter inside. Okay. Subarine depositions will make up the Casparian strips. Casparian strips. Okay, subarine depositions. Now, there is a line of NCERT. Subarine depositions are tangentially and radially. Tangentially and radially, we can see subarine depositions. Let me show you how. All of you can see this? Can you see? This is a typical barrel shaped cell. This is called as your transwall. On the top of the transwall, we can see subarine depositions. And also radial wall. This is radially, tangentially, radially, there is depositions of what? Depositions of subarine called as Casparian strips. Casparian strips are running tangentially, radially. Casparian strips are running. Understand, I know many would have by-hearted this part. I am asking you to understand today. Okay. So, Casparian strips are running transverse, longitude and radial wall. Now, you will ask me, sir, why is it not present throughout? Students, understand, if subarine was there completely filling up, can water even enter inside? Can the root even do conduction of water then? No. So that is why very smartly, the subarine depositions is only found in the radial and tangential wall, not throughout. Okay. Now, where were we now, students? Where were we? We finished something called as your epidermis, done, cortex, done, endodermis is done. Innermost layer, intercellular space is absent here, parenchyma, checkpoint, cells are barrel shaped with subarine depositions making up the Casparian strips. Any doubt till now? Quickly ask me. Any doubt till now, students? If there is no doubt, tell me what is the next layer now? If you look at the diagram, you can see the next layer here. Pericycle. Pericycle. Now students, tell me, is pericycle a single layer or multi-layer now? Look at the diagram and tell me. Look at the diagram, all of you. I can see 1, 2, 3. Can I say few layer? Yes. So pericycle is going to be few layer. Students, trust me, this is very fun. It is so fun to do this. And trust me, once you have mastered right, when you look at the diagram and write, you will also enjoy this process. Okay, this is always fun for me. I don't know why. It's few layer. Now, in pericycle, students, if you look at the pericycle, it is also made up of parenchyma. Now, your pericycle has two main roles here. The cycle has two main roles here. The first role is that lateral roots, not root hair. Root hair is different. Lateral roots. What are lateral roots? For example, this is the main taproot, right? For example, this is the main taproot. In this main taproot, this is called as primary root. Primary root. From this main taproot, from the inside, we can see the development of secondary root. This secondary root is also called as your lateral root. And where is the lateral root originating from? This lateral root is coming from where? It is coming from the pericycle. It is coming from the pericycle. Okay. So lateral roots originate from here. Lateral roots origin is happening here. Origin is happening in the lateral roots. And also pericycle is also involved in secondary growth. Also involved in secondary growth to some extent. 1/3 of vascular KBM. Done. Pericycle is done. Now students, only 4 points. See, only 4 points. Now if you go even more inside, what do you find, students? Now what inside, what do we have? Inside pericycle we have the vascular bundles. Now we have the VB. We have the VB inside. Vascular bundles. We have the vascular bundles. Now tell me, students, it is the root. It is a root sectioning. Will it have radial arrangement or will it have conjoint arrangement? Think back, all of you. I will only show you. It is a root. It is a root. Will it have? Where did it go? Will it have? Where did it go? Stupid. It's here. In radial arrangement, I wrote roots. Conjoint, stem and leaf. So radial is what? Radial is going to be root. Radial is going to be root. Radial arrangement. Radial arrangement. Now, what is the protoxylem? Protoxylem and metaxylem. I always told you RX. Root X arch. So can I tell we have the X arch here? We have the X arch condition also here, where protoxylem is towards the outside. Protoxylem is towards the outside. Metaxylem is towards the inside. Yes, metaxylem is towards the inside. X arch condition. X arch condition. And here vascular bundles are a little patchy. Patchy vascular bundles. Patchy. Now count the vascular bundles now, students. Count the vascular bundles. 1, 2, 3, 4. 4. So usually it varies from 2 to 4. It varies. 2 to 4 vascular bundles. And it is called as dyad or tetrad. Tetrad condition. Dyad or tetrad condition. Dyad or tetrad condition is vascular bundles here. Vascular bundles here. Okay. Oof. Next, what do we have? What do we have? Next is gonna be a pith. Pith. Here, middle part. Pith. Next one is gonna be pith. Pith is very small here. Very small. I can also call it inconspicuous. Inconspicuous pith. Very small pith here. Very small inconspicuous pith. Then we have the entire thing is called a stele. Entire thing is called a stele. What is stele? Stele is basically the entire thing. Pericycle plus the vascular bundles plus the pith. That is pericycle plus vascular bundles plus the middle path. Pith is called as the stele. It is called as the stele. It is called as the stele. Okay. And between xylem and phloem, this is your xylem phloem. Between xylem and phloem, between your xylem and phloem, there is something called as conjunctive tissues. Conjunctive tissues are present between xylem and phloem. That's all. Over. That's all over, students. I'll give you a small recap. Understand. Only 4 slides. First one is epidermis. Listen to me very carefully. First of all, first one is called as epidermis. Outermost layer, single layer, root hair, protection, parenchyma, intercell space is absent, cuticle absent, stomata absent. Epidermis done. Epiblema, also known as epiblema. Then we have cortex, many or multi-layer, parenchyma, ICS is present, intercellular space is present. Then we have endodermis, innermost layer, single layer, intercellular space is absent here, parenchyma, checkpoint, superior depositions, aspirant strips. Then we have pericycle, few layer, parenchyma, lateral roots originate from here and also responsible for secondary growth. Then we have vascular bundles, radial, exarch, patchy, 2 to 4, done. Pith is very tiny. Stele is what? Stele is nothing but your pericycle plus vascular bundles plus pith. In the between xylem and phloem, we have the conjunctive tissue. Done, that's all. That's all is the entire Dicot root. Let me show you proof. Read the lines of NCERT, all of you. Read the lines of NCERT. Time starts now, go. Outermost layer is called as epiblema. I told you. Also called as epidermis is called as epiblema. Done. Protrude in the form of unicellular root hair, appendages. Done. Cortex consisting of several thin-walled parenchyma cells. So several means what? Many. I told you many for cortex. Yes. With ICS is present here. Innermost layer of the cortex is called as endodermis. Barrel-shaped cells. Without any ICS in endodermis. The tangential as well as radial wall of the endodermis, radial walls of the endodermis have depositions of the water impermeable. Water cannot enter there. Waxy material called as suberin, done. In the form of aspirant strips, done. Next to few layer, we have few layer. We wrote few layer of what? Parenchymatous pericycle, done. What happens here? Initiation of lateral roots, origin of lateral roots, and also vascular cambium during secondary growth. I told you, also vascular cambium during secondary growth. Pith is small and inconspicuous. I told you, pith is very tiny pith. Okay. The parenchyma cells lie between the xylem and phloem, conjunctive tissue. Between xylem and phloem, conjunctive tissues. How many xylem and phloem? 2 to 4. Dyad or tetrad? Xylem and phloem. Dyad or tetrad? Patches. It's patchy. I told you patchy. Later, this is secondary growth. Later, cambium ring develops between the xylem and phloem. Cambium ring, I have told you this already. In the interfascicular cambium and intrafascicular cambium, because the cambium ring is developing. We learned it in the meristem, lateral meristem. All the tissues on the inner side of the endodermis, that is pericycle plus vascular bundles plus pith, is making up the stele. Towards the end I told you. Now you tell me, students, isn't the entire structure of your Dicot root very simple? Every single point did we cover? Actually, we wrote more points. Here. All this is not given. I have written extra points for you to understand Dicot, all this, because they'll ask an examination. Few layer parenchyma, absent, very important line. Intercellular space is absent here. Checkpoint, suberin, patchy. Epidermis, cortex. Done. That's all. Is your Dicot root. If you understood Dicot root, I can teach you monocot root in 1 minute. I can teach you entire monocot in 1 minute. That's all. Very simple. Now monocot you understand. Monocot root you understand, students. Monocot root you understand. In the case of monocot root, epidermis, cortex and pericycle is same to same as your, just like your Dicot, same to same. The only difference will come major in the middle part. That is the vascular bundle major difference. Now it is the root. Tell me, exarch or endarch? It is a root. We will have exarch. Yes, exarch condition. Exarch. Radial, same. Radial. But it is the monocot root. Monocot root. KMBM is going to be absent here. So can I call it closed also? It's closed. That was open. Okay, that was open. Closed here. Okay. Why? So if it is closed, secondary growth is absent. Secondary growth is absent. Secondary growth is absent. That's all. Apart from that, the last major point where many questions come from is going to be look at the vascular bundles. How many do we have? Xylem bundles. 1, 2, 3, 4, 5, 6, 7, 8. So can I tell vascular bundles is more than 6? More than 6 here. Such a condition where vascular bundles are more than 6 is called as polyarch condition. Polyarch condition. Polyarch condition. Done. Apart from that, look at the pith. Here it is not visible, but pith is very large here. Pith is large, made up of parenchyma. Made up of parenchyma. Made up of parenchyma. Pith is large, well developed. Pith is here. Done. That's all. It took me 1 minute. It took me 1 minute to teach you monocot root. See here. Anatomy of monocot root is similar to dicot root in many ways. It has epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As compared to dicot root, which has a fewer xylem bundles, there is usually more than 6 polyarch condition here. Polyarch condition. Pith is very large and well developed. Monocot root do not undergo secondary growth. It is closed. Vascular bundles are closed here. Closed condition. Closed. Done. That's all. Done. Look at this question, students. Don't worry, all these notes will be given to you. A transverse section of monocot root shows the following internal structure organization. Arrange them in a correct sequence starting from periphery to the center. From periphery towards the center. Tell me which one will be in the there in the outer part. First we have epidermis, always epidermis. C will come first, epidermis. Then we have the cortex. Yes, cortex. Then we have the first we have epidermis. First we have the epidermis. Then we have the cortex. Innermost cortex is called as endodermis. Then we have pericycle, and finally we have the pith. We have the pith here. Okay. See here, epidermis, cortex, endodermis, pericycle and pith. A transverse section of plant part shows polyarch condition. See, answer is given here only for you. Polyarch condition. NEET 2023 question. Polyarch condition, radial, exarch xylem with endodermis and pericycle. Plant part is identified as. Students, radial and exarch, it is a root. It is a root, pakka root. Okay. Radial and exarch. But polyarch condition, that is more than 6 xylem bundles. Where do you find that? Monocot or dicot root? Monocot or dicot root? Tell me. It is going to be your monocot root. Monocot root. Done. Initiation of lateral roots. Initiation of lateral roots and vascular cambium during secondary growth takes place in. I told you, lateral roots are coming from where? Theory and NEET is almost same to same. It is only the approach. See, you learn the theory with me only. For NEET, you need to know the what is that? You need to have a proper time management and the quick thinking skills to point the answers here. See here, I told you lateral roots and vascular cambium. Your mind should go to pericycle directly. Pericycle. Okay, pericycle directly. Okay. In dicot root, vascular bundles originate from. Vascular bundles, vascular cambium originate from. Where does it originate from? Oh, this is This is secondary growth. This is removed from your syllabus. Don't worry. It is part of secondary growth. That question is secondary growth, not for you. Okay, that chapter part is deleted. Caspian strips occur in. Where do you find Caspian strips, students? Do you find it in epidermis, pericycle, cortex, or endodermis? In endodermis, there is something called as suberin depositions, which is making Caspian strips. Endodermis. Endodermis. Okay. That's all. Now you go find any other question also. Go find any other question and try solving. It becomes very easy for you. Okay. Go try solving any other question. It becomes very, very easy for you to solve the entire root part here. Monocot or dicot. It becomes very easy for you. So with that being said, now students, can we go to the next section that's called as your dicot stem? Can we start? Yes or no? Minecraft, Prem, Madhumiti, Sukanya, Pooja, Prem. Can we start now, students? Can we start the next part that is going to be dicot stem? Yes or no? Issue? Show some energy in the chat now. I think it's been 4 hours. It's going to be 4 hours, I guess. 6, 7, 30, 8, 30, 9, 10, 30. Yes, 4 hours. Almost 4 hours, right? Almost 4 hours. Now, we will start with the dicot stem now. We will start with the dicot stem now, students. Right? Can we start? Dicot stem. Now, tell me, students, which is the outermost layer and what do you see in the diagram? All of you go. Which is the outermost layer? Tell me, which is the outermost layer right here and what do you see? Outermost layer. So, I can confidently tell the outermost layer here is the epidermis, and all of you will be able to tell me that, yes sir. Outermost layer is the epidermis here. Epidermis. Now students, the epidermis is the outermost layer. Do you find as a single layer or multiple layers? Tell me, is epidermis a single layer or multiple layer? Tell me. Similarly, all of you know, epidermis is single layer, outermost layer, protection, all that is very common points. All that is very common points. Okay. Apart from your common points, do you find any type of appendage? Do you find any form of appendage? Epidermal appendage here. Like, yes sir, I can see the trichomes here. Can you see epidermal hair? Trichomes. I can see the trichomes here. I can see trichomes here. Now it's a stem, right? Can we find stomata here? Stomata can be present in some species. In some species, stomata is absent. What about cuticle? Cuticle may be present or it can be absent in many cases. Or it can be absent in many cases. Okay, it can be absent in many cases. Okay, that's all. That's all is epidermis. That's all is epidermis. Okay. Single layer. Single layer. Done. Epidermis done and dusted. That's all. Single layer. Okay. Oh, wait. The main point we didn't tell only. Students, in epidermis, do you find the common thing? Do you find interstellar spaces here? Do you find interstellar spaces? Interstellar spaces. Epidermis, no. Compactly arranged, no. Compactly arranged. Epidermis, no interstellar spaces at all. Okay. Now, second layer, students. Second layer. Second layer is called as your hypodermis. Can you see here? It is called as your hypodermis is made up of what? Hypodermis is made up of 𝐂𝐡𝐞𝐤. All of you remember this part, right? It is made up of 𝐂𝐡𝐞𝐤. All of you remember 𝐂𝐡𝐞𝐤. And students, tell me, where do you find 𝐂𝐡𝐞𝐤? Where do you find 𝐂𝐡𝐞𝐤, students? 𝐂𝐡𝐞𝐤 is mainly found in the case of stem or petiole. Stem or petiole. Okay. Do you find any type of interstellar space here? It's compactly arranged, no? Compactly arranged. Interstellar space is completely absent here. What is the main function here? Function is mechanical strength. Mechanical strength. Can it do photosynthesis? Yes, it can do photosynthesis. It can do photosynthesis is also possible. Photosynthesis is also possible here. Beautiful. Done. Now next layer you see. Next layer is the cortex. Next layer is the cortex here. Cortex. Now students, small twist. Cortex is made up of 3 different layers. The first one is actually the hypodermis. First one is actually the hypodermis. Second one is called as a cortical layer. Is called as your cortical layer. Now students, tell me, what is cortical layer made up of? Can you see it is green color here? Can you see the cortical layer is made up of something? Can you see? Cortical layer is made up of your parenchyma. It is made up of parenchyma. We can find some form of intercellular space here. Some form of intercellular space. Can you see here, here? Intercellular space is present here. Intercellular space is present here to some extent. Single layer? No, not single layer, few layer. No. Innermost layer of cortex. Innermost layer of cortex is called as your endodermis. Is called as your endodermis. Now students, tell me, is endodermis a single layer or multiple layer? We can find only single layer, but under endodermis, there is one point which they keep on asking all the time. One point for endodermis, they keep on asking all the time. What is endodermis is known for? See, for example, in the case of root, endodermis was known for superinterpositions and Caspian strips. But in the case of stem, the dicot stem mainly, it has something called as your starch sheets. Starch sheets. That's all you need to remember. That's all you need to remember about your endodermis. Okay, endodermis. Now students, next one. Next layer is called the pericycle. Next layer is called as pericycle. Pericycle. Now students, if you look at the pericycle here, can you see the pericycle a little wavy? Wavy. Can you see? Pericycle is wavy here. Pericycle is wavy. It is very wavy in nature. Wavy in nature. Now look at the shape of this. See, all of you know, this is a full moon. This is a half moon. And half moon is also called as semi-lunar. Full moon is called as lunar. Full moon is called as lunar. And half moon is called as semi-lunar. Okay. The shape here is semi-lunar shape. Semi-lunar shape. I will show you the questions. Questions are there. Don't worry, questions are there. You will know exactly which type of question they will ask you and how you can answer those. Okay. Semi-lunar shape, and it is made up of your sclerenchyma. How do I remember? Diagram. Brown. Sclerenchyma. Made up of sclerenchyma. It is made up of sclerenchyma. Done. Pericycle is also done. Now finally we come to the vascular bundles. Friends, this is a stem right here. This is a section of a stem. This right here is a section of a stem. Tell me, will it have radial arrangement or conjoint arrangement? Tell me, all of you. Will it have a radial arrangement or a conjoint arrangement? All of you tell me. All of you tell me. Will it have a radial arrangement or it will have a conjoint arrangement? Go. The first main point about your vascular bundles are arranged in a ring structure here. Ring arrangement is here. Yes, ring arrangement is here. Apart from the ring arrangement, It is going to be your conjoint. The present to it's a stem. It'll have collateral. It'll have end arch, and finally it has cambium here. Can you see cambium is present here? Cambium is present. Between xylem and phloem, can you see cambium here? Vascular bundles, we can see cambium here. Okay. So if cambium is present, can I call it open? Yes, sir. I can also call it open. I can call it open. Okay. So let's see one more time if we missed any point here. Ring arrangement is there. Conjoint, collateral, and dark, as well as your open. Okay. Done. Diagram, one point I'm missing diagram here. You see the diagram? Can you find middle rays here? Middle rays can be found here. Where are they present? Between vascular bundles. Between the vascular bundles, we can see the medullary rays, and it is the same medullary rays. It is the same medullary rays which will get converted to de-differentiated to the interfascicular cambium later on. Interfascicular cambium. Okay. Last, I think, is pith. Middle part is the pith here. This one, pith here. Pith is going to be like a large pith. Large pith. At the same time, pith is made of parenchyma. And also interstellar space is present here. This small spaces are present here. Okay. Now understand what we just did. I'll give you a small recap. Few points, 2 to 3 points everywhere. Okay. First of all, it's epidermis, single layer, done. Epidermis, single layer, done. Then we learned about trichomes. Yes. Stomata can be present, can be absent. Cuticle may be present, may be absent. Interstellar space is completely absent. Epidermis, done. Then we have the hypodermis. Collenchyma, all of you know, absent completely. Mechanical photosynthesis it can do. Next is the middle layer, that is cortex. 3 subzones. Hypodermis, done. Cortical region, parenchyma, few layer, interstellar space is present here. Endodermis, star sheet. Cortex, done. Pericycle. Pericycle is wavy, semilunar shape, sclerenchyma. Okay, sclerenchyma is present here. Then we have the vascular bundles. Ring arrangement, conjoint, collateral, endarch, and open, as well as open. See, your endarch means what? Protoxylem is towards the pith here. Protoxylem is towards the pith. Can you see? Towards the pith. Metaxylem away from the pith. Pith, large, parenchyma, interstellar space is present here. Okay. Now you read the NCERT and tell me if you did not understand any point, then we will go ahead. Okay. So tell me if you don't understand any point anywhere. Epidermis is the outermost layer. Protective, I told you, always protective in function. Thin cuticle. Cuticle is present here, or may be present, may not be present. Trichomes is there. Okay. Few stomata can be present. It can be present. Okay. The cells arranged in multiple layer between epidermis and pericycle constitute the cortex. What is cortex? Epidermis and pericycle. Between that is called as the cortex here. Okay. It consists of 3 subzones. The outermost is called as hypodermis, I already told you. Made up of collenchyma. Okay. Function, mechanical strength to the young stem. Then cortical layer. The second one is called as cortical layer below the hypodermis. Below the hypodermis is called as cortical layer. Now consisting of round, thin-walled parenchyma cells, we did that. Conspicuous interstellar space is present. Interstellar space is present here in the cortical layer. Then we have the innermost layer of the cortex that is called as endodermis. What is so special about it? Star sheet. Star sheet is a specialty about it. Then we have pericycle, semilunar shape. semilunar shape of pericycle. Semilunar patches of sclerenchyma. See, I told you, sclerenchyma, brown color. Okay. Between the vascular bundles, we have the medullary race. Medullary race between the vascular bundles. Vascular bundles are arranged in a ring. Ring arrangement of vascular bundles of a dicot stem. It is going to be conjoined, open with endark protoxylem. Endark condition done. Okay. Large rounded parenchyma, cells with large intercellular spaces, which center position, that is called as the pith. So pith is what? Pith is very large, parenchyma, intercellular spaces. Did you do that? Did we do that? It's going to be large parenchymasis and intercellular spaces present here. ICS is present here for the pith. That's all is going to be your dicot stem. That is going to be your entire dicot stem. Understood, all of you? Any doubt anywhere? See, students, it is very easy. It is very easy to learn anatomy. Whatever you see, you write down. Whatever you see on the diagram, write down and then you learn. It's very, very easy to understand. Now, if you understood the dicot, dicot stem, monocot stem is even more easy. Monocot stem is even more easy. Okay. Now, what do you see? Tell me again here. Tell me again, what do you see here? Again, we have the epidermis here, students. Again, we have the one second, students. Let me tie my shoelace. 1 second. Shoelace is coming out. Okay. Now, what do you see here, students? We see epidermis here. Okay, we see epidermis. And we see the epidermis. Same, epidermis is going to remain the same itself. Epidermis is going to remain the same itself. Epidermis, outermost layer, protection, parenchyma, same rules, nothing different. But what is going to be different here is the hypodermis. Why? Because didn't I teach you in today's class, your collenchyma is only present in the case of your dicot stem and leaf. Dicot stem and leaf collenchyma. But did I also tell you that collenchyma is absent in the case of monocots? Absent in the case of monocots, collenchyma. So can I tell right now, the hypodermis is made up of sclerenchyma because it needs support. It needs support, right? You can't do parenchyma here. You can't add parenchyma here. Not possible. Doesn't give structural stability. So you need to add sclerenchyma here. Now, if you look at the cortex, students, the ground tissue, do you see any type of subdivisions here? No. If you look at the ground tissue here or the cortex is not differentiated. Cortex is not differentiated. Completely scattered. Can you see here? All the vascular bundles are completely scattered everywhere. Now also, I was teaching you about phloem parenchyma that time. I told you, phloem parenchyma, in the phloem we learned it. Quick reminder, I told you PYQ also. Phloem parenchyma is absent here. This is the most important line which everyone ignores throughout the studying part. So please note down this. Phloem parenchyma is absent here. Phloem parenchyma is completely absent here. Okay, completely absent. Did we miss anything? Vascular bundles. Okay, vascular bundles. We'll come back to vascular bundles now. Okay, now we study the vascular bundles, students. If you notice the vascular bundles, you will notice a small trend here. If you notice the vascular bundles on the periphery, we have small vascular bundles. In the periphery, we have vascular bundles are small, but many in number. Can you see here in the periphery? Periphery, you can see here, all the vascular bundles are many in number, but tiny, tiny. Towards the center, if you go, vascular bundles are few. You can see here, very few in the center, very few. You can count the center, but large. But they're completely large here. Okay, but completely large towards the center. Are we missing any point about water cavities? I can tell you water cavities later on. Are we missing any point here? Vascular bundles are covered, right? Yes, vascular bundles are completely covered. Bundle sheet. If this is a vascular bundle, students, if this is a vascular bundle, this is a vascular bundle, it is completely covered by a layer around it. A layer is called as what? Bundle sheet. Bundle sheet. And it is here it is made up of sclerenchyma. Let me check it is given in your NCERT or not. I keep remembering this. Okay. Now apart from that, do you see this water droplets anywhere in the middle? Water cavities. Can you see here also? Round blue structures here. Blue color structures. Yes. You can here also you can see. Okay. So the last point which I have to tell you here is presence of water cavity. Presence of water cavity. Okay, presence of water cavity. Okay, skeleton head shape. Exactly, it's called a skeleton head shape. And students, do you remember in the starting of this? I showed you this. Can you see here? Once I'll show you this. 134. You have to come back. No. Friends, do you see this structure? Skeleton head shape. And this is a section, my section. This is my section. Okay, this is my sections. I took this section under the microscope. Okay. Done. So in monocot stem, nothing much there, very simple it is. Monocot stem is very simple. Okay. Monocot stem is very simple here. Monocot has a sclerenchyma hypodermis, I told you already. Large number of scattered vascular bundles. Scattered vascular bundles. It means they are not very differentiated. They are separated everywhere. Separated everywhere. Vascular bundles. Surrounded by, see, oh, it's given. Sclerenchymatous bundle sheet. So bundle sheet is present. Bundle sheet is present around the vascular bundles. Okay. Vascular bundles are conjoined, closed. Oh, we didn't write at that point. Huh? Conjoined and closed. Vascular bundles are going to be conjoined because it is a stem and closed. Why closed? Because it is a monocot. That is why it is going to be completely closed. Conjoined and closed. Clear all of you? Clear? Conjoined and closed. That point was missing, I guess. Conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent. Very important line of NCERT. Is absent. Phloem parenchyma is absent. Water containing cavities are present within the vascular bundles. Water containing cavities are there. Small cavities are there called as water content cavities. We wrote that also here, right? Water containing cavities. Water cavities are present. Water cavities are present here. Okay. So how will they ask you questions? Very good question. See this question now. A major characteristic of monocot root is the presence of. When they ask you such questions, you should always go for the most predominant reason, the main correct reason. Now that is going to be your, see, this can also be correct. Oh, this is root, right? Root. That is going to be vascular without cambium. So So cambium is absent in the case of monocot. In the case of monocot, it is going to be absent. In the case of monocot, vascular bundle is absent here. Does not have cambium. Interfascicular cambium develops from the cells of. I've told this repeatedly today. I've told you repeatedly today. Okay. It is going to be 3 only, no? See here. 3 only. Why 3 only? Because cambium should not have any type of, cambium should not be present in monocot, right? Monocot should not have any cambium. So I've told you already this. Your interfascicular cambium is developing from the medullary rays. Developing from the medullary rays. Medullary rays undergoes dedifferentiation to make the interfascicular cambium. Very good question. See this. Water containing cavities in vascular bundles are found in. Students. Sir, how is secondary growth in? The question says monocot. The question says monocot. And in the case of monocot, secondary growth is completely absent. Completely absent. So cambium without cambium, right? Without cambium. This question is very nice question actually. This question is very nice question. It says water containing cavities. We just now learned in the case of monocot. In the case of monocot, stem it was. I cannot tell you more than that. I cannot tell you more than that. That's the major clue. That is the major clue. Okay. Sunflower is Solanaceae. Sunflower is going to be your Astraceae, Dicot. Maize, Cycas is gymnosperm. Pinus is also going to be a gymnosperm. But maize, students, maize is a monocot. Maize is a monocot. This is a dicot. These two are gymnosperms. Right? So water containing cavities. Water containing cavities, where do you find? In the case of maize, maize is the monocot, grasses, poaceae, right? So maize is the answer. Very good question, very good question. Okay. So with that, we come towards the last topic for today. Last topic, very cute topic, very easy topic to understand. That is going to be your leaf. Can we start, students? Can we start the last topic for today, which will take me around 10 minutes or 15 minutes to maximum 15 minutes to teach you. Very simple, very easy to understand. Okay, leaf. Understand the leaf structure, students. Understand the structure of leaf. This is a typical structure of leaf. This is a typical structure of a leaf. Okay, it has veins and veinlets. Largest vein is called as your midrib. It's called as midrib. Here we have something called as the epidermis. Here also on top also we have the epidermis. Epidermis on top. Now the difference is it is called as separate names. Upper epidermis and lower epidermis. For example, this is the lower epidermis and this is the upper epidermis. Upper epidermis. And remember, students, the upper epidermis is also called as your ventral or adaxial side. It is also called as your ventral side and adaxial side. Also called as adaxial side. Now, what about the below one? The below one is called as a dorsal or abaxial. So remember, students, back door. Okay. So it's called as dorsal or abaxial, backside. Lower back. Okay. It is called as abaxial side. Abaxial. Okay. It is called as abaxial. Called as abaxial side. Okay, adaxial and abaxial side. Okay. Now here in the mid, this is called as midrib. It's called as your midrib in the middle. Midrib in the middle. Beautifully done. Now students, you need to understand something here is something called as the dicot leaf and the monocot leaf. Questions will directly come from the labeling, or why is it called as dicot and why is it called as monocot. There are separate names here. Question will come directly from there. Or other than that, there is a small concept called as bulliform cells. Question will come from there, nothing more than that. Okay. Now understand, why do we call that? Why do we call your dicot leaf as dorsiventral leaf? Understand. We call this leaf, section of a leaf, called as dorsiventral. Why do we call it dorsiventral? Dorsal side and ventral side. In the dorsal side and ventral side, we have two different types of tissues. In the dorsal side and ventral side, we have two different types of tissue. That is why it is called as dorsiventral leaf. On the ventral side, we have palisade mesophyll. On the dorsal side, we have the spongy mesophyll. Understanding? That's why it's called as dorsiventral leaf. Dorsal side, ventral side, we have, ventral side and dorsal side, back side, not dorsal. Dorsal side and ventral side, we have different types of tissues, dorsiventral. At the same time, monocot leaf, students, monocot leaf is called as isobilateral. Iso means same. Bi means both the sides. It is same. So isobilateral sites we have what? In both the sides we only and only have the mesophyll cells. It could be dorsal side or ventral side. Dorsal or ventral, we only have same type of cell, mesophyll cell. So it's called as iso, means same, bilateral, both the sides. Okay. Now if you look at your dorsiventral leaf, it has something called as your palisade mesophyll on the top, spongy mesophyll on the bottom. Palisade mesophyll is arranged parallelly with respect to each other. See, it is arranged parallelly completely, palisade mesophyll. On the bottom we have the spongy mesophyll. Spongy mesophyll, all of you remember the Ehrenheimer buoyancy, right? So spongy mesophyll on the bottom, which can help in gas exchange. Now in the case of your dicot leaf, students, in the case of dicot leaf, what happens is on the above side, usually we have a thick cuticle on the upper side, and on the below side we find more stomata. In the case of below side. Why? Structure, you understand, structure. This is a particular stem. On the stem we have a leaf like this. We have a leaf on the stem. All the sunlight is falling on this. Leaf. Now if sunlight is falling on this, imagine if we have a stomata on top. If you have stomata here, it will obviously close off. No use, right? So if we have more stomata in the bottom, it is much more useful. So we have more stomata in the bottom here. Okay, and also remember, even the vascular bundles, the spreading of vascular bundles in the midrib region. In the midrib region, you will find more vascular bundles compared to the other veins. And remember, all the vascular bundles are present in the veins only. Mostly in the veins, because with the help of veins there can be transfer of water, conduction of water can happen. Okay. See what is given here, students. Very simple. The ventral section of a dorsiventral leaf through a lamina shows three main parts. Epidermis, outermost layer, mesophyll is the middle part, and vascular system. Okay. Epidermis, which covers both the upper surface and lower surface. Adaxial, abaxial. Adaxial top, abaxial bottom. Okay, bottom. Conspicuous cuticle. Cuticle is present, I told you. The abaxial epidermis generally bears more stomata. The below surface has more stomata than the adaxial. The latter even lacks stomata because upper will be exposed to sunlight now. That we have mesophyll cell. Mesophyll is divided into palisade parenchyma and spongy parenchyma. Your contains chloroplast and carry out the photosynthesis. It is made up of the parenchyma, two types of cells, palisade parenchyma and spongy parenchyma. So dorsiventral leaf, dorsiventral leaf. The adaxial placed palisade parenchyma on the top, adaxial placed parenchyma is made up of elongated cells. I told you, elongated cells which are arranged vertically and parallel to each other. Can you see here? They are arranged elongated cells which are arranged vertically. Elongated cells arranged vertically and parallel to one another. Parallel to one another. Like how you put biscuits, no? One nice biscuit you added. Nice biscuit, one after two. Biscuits like it is added. Bakery biscuit. Okay. And arranged vertically and parallel to each other. The oval and round, which is the oval and round is the spongy parenchyma. Loosely arranged spongy parenchyma, oval and loosely arranged. Can you see here? It is oval and loosely arranged. Oval and loosely arranged. That is spongy parenchyma. Okay. Numerous large spaces, air cavities are present between these cells. Vascular system includes vascular bundles, obviously, which can be seen in the veins and midribs. I told you, all the vascular bundles are present where? They are present in the veins and the midribs. The size of the vascular bundles are dependent on the size of the veins. Larger the vein, larger the vascular bundle. Okay. The veins vary in thickness in reticulate venation, obviously, in Dicot leaf. The vascular bundles are surrounded by a layer of bundle sheet here. Again, so in your case of your monocot stem, we had the bundle sheet covering, no, monocot stem, sclerenchyma. Here also we have bundle sheet, but it is parenchyma. I don't think it's given here, that's all. Okay. So main points I've told you everywhere. Very simple. Leaf is very simple. Okay. Monocot leaf also very same thing. In the case of monocot leaf, understand students, in the case of monocot leaf, mesophyll present is throughout. But if you look at the structure of a typical monocot leaf, on the stem, a leaf is almost parallel. Leaf is almost parallel to the surface. So what happens is here, even if the sunlight is falling, it doesn't matter where it is falling, because since it is almost parallel, or even sometimes it is pointed upwards, it doesn't matter the number of stomata in the location, because in the case of your monocot leaf, the stomata is equally arranged. You can find stomata on the top and stomata on the bottom in equal number, here, in the case of monocot leaf. Okay. See. Isobilateral leaf, all of you know why it's called isobilateral. Stomata are present in both the surface epidermis. Okay. Mesophyll is not differentiated into spongy and palisade. There is no differentiation. We have spongy, we have your palisade. Differentiation is not there. Differentiation is only there in the case of dicot. Okay. Now, on something called as bulliform cells, understand bulliform cells. Very important concept. Many questions have come from here. So, bulliform cells are basically large, colorless, empty cells. They are large, colorless, empty cells, right? Water can enter inside. Every single time water is entering inside, the leaf is aram se open. Okay, it is open like this, leaf is open. But as soon as the water is removed, if the water is removed, the leaf will stop curling. Not like this. The leaf is something like this, right? Leaf will curl like this. As soon as the water is removed, the leaf will start to curl inwards. It will curl inwards. Okay. In grasses, that's monocots, certain adaxial epidermal cells on the top, epidermal cells, veins modify themselves into large, empty, colorless cells, bulliform cells. When bulliform cells in the leaf have absorbed water, it has water, it is turgid. Water, it has turgid. So as soon as the water is lost, all the leaf will show curling on the inside. Okay. When they are flaccid due to the water stress, they make the leaves curl. They make the leaf curl towards the inward to minimize the water loss. The parallel venation in monocot leaf reflect the near similar size of the vascular bundles except in the main stem. So vascular bundles are same size here. Why? Because the veins are same size. Larger the vein, larger is the size of vascular bundle. Okay. Beautiful. See this question, all of you. See this question. See this question, all of you. How many questions are there? Let me just check. How many are there? Don't worry, time is there. Okay. You are supposed to choose the correct answer. Choose the correct answer. Okay. In root, xylem and phloem are in the vascular bundles are arranged in an alternating manner along different radii. Yes, that is radial arrangement. True. Conjoined closed vascular bundles do not possess cambium. Correct. Because it is closed. Closed means no cambium. Correct statement. In open vascular bundles, cambium is present between xylem and phloem. Yes, open is present. Vascular bundles of dicot stem possess endark protoxylem. Yes, it is stem endark. All of you know that. In monocot root, usually there are more than 6 xylem bundles. That is basically polyarch condition. Polyarch condition. So this is also correct. So how many are correct, students? How many are correct? All are correct now. All are correct. All are correct. See this question. Large empty colorless cells of adaxial epidermis along the veins of the grass is called as your bulliform cells. Bulliform cells. Okay. Bulliform cells are responsible for what? They are responsible for, how many questions are there? Inward curling, right? All of you know inward curling. Inward curling in monocot. Now, this is a very good question. This is a very good question, students. Very good question. Okay. Very good question. This is your homework question. I'll give you some hints. This is your homework question. I'll give you small hints, okay? I'll give you small hints. Okay. Large number of scattered vascular bundles surrounding the bundle sheet. So scattered vascular bundles, we read this term somewhere. Okay. Large conspicuous parenchyma ground tissue. Vascular bundles are conjoined and closed. If it is closed, students, if it is closed, it means cambium is absent. Cambium is going to be absent. Can it be dicot now? Can it be dicot? It cannot be a dicot stem, or it cannot be a dicot. It cannot be a dicot stem. It cannot be a dicot root because they are open. They are open. So, can I remove dicot stem and dicot root? And also it says conjoined. Conjoined. It is only found in the case of stem. So, root is automatically gone. So, I hope I almost told the answer only, but let me know in the comment section what is the answer. So, this is how they'll ask you questions. This is how they'll ask you questions, students. This is how they'll ask you questions for this entire chapter. This is how they'll ask you questions. So, homework question, let me know in the comment section. And with that, the entire chapter is over. It's actually over, done and dusted. I want everyone to comment in today's video, anatomy done and dusted. Okay, anatomy is going to be done and dusted. Done and dusted. Now students, all of you know, right? All of you know, NCERT exam is coming nearby. So if you haven't, registered for NCERT exam, register right now. NCERT exam. Okay. Register NCERT exam, write the NCERT exam, a lot of benefits, and you will see the benefits, students. So thank you so much for today's class. I saw so many new students today. Diksha is new. No, old. Yeshu. So I saw many new faces today. Students, all the students who are new to the channel, subscribe to the channel, smash the like button, and also let me know in the comment section how was today's class, because this is a beautiful chapter. It's a very beautiful chapter. Okay, very beautiful chapter. The only part is you don't learn it in a proper way. So what happens is, technically you usually tend to by heart this chapter. There's nothing to by heart here. It is all about experimenting and learning. And trust me, it would have been best if I could teach you offline and sitting in front of you showing you the sectioning. Since that is not possible, all of you need to learn the diagrams, See the diagram, write information. See the diagram, write the information. That's how you learn this chapter. Okay. So thank you so much, students, and I will see you in the next class. And bye bye, all of you. Take care. Okay. You want the notes? No, I will send you the notes. Don't worry. Notes are there coming on the Telegram channel. Okay. Baswaraj sir, biology. Okay. Bye bye, all of you. Take care. Have a great day ahead.